Okay, so welcome everyone. My name is Ellie Andrews, and I'm the UC Cooperative Extension Specialty Crops Advisor for Sonoma, Marin, and Napa counties, and I work with a lot of apple growers here. So I'm really excited to host this UCCE Apple webinar today. We have an awesome lineup. I'm really appreciative to all of our speakers here on this flyer, which I think everyone here who registered has seen this flyer, but this is just a breakdown. We're gonna have half-hour segments. We'll have a break halfway through. It's going to be great. You can take a break at any point in time. I will be recording this and sending out the recording to everybody registered. So, if you want to go back and rewatch some parts or see a part that you missed, you're welcome to do that. And so we're going to get started with Akif Escalon talking about fungal disease ID and organic management. So I'll hand it over to you, Akif.
Thank you very much, Ellie, for organizing this meeting and for your invitation. So today, let me introduce myself first. Some of the apple growers may not know me. I am extension specialist working on primarily fungal disease of plum fruits, including apple, pear, and other plum fruits as well in California, and also as well as grapevine and other wine crop diseases in California. So with that, thank you very much for the invitation. This is an opportunity for us to share some of the information that we have been working on apple and pear diseases. Today, I'm going to focus on fungal diseases causing branch dieback, wood canker, and blotch diseases on the leaf and fruit of apple in California. These are the common diseases that we have identified so far. So before I start, I would like to introduce you the extension specialist role in the California based on the crops that I cover. So we basically work with the farm advisors. They are like Ali, UC Cooperative Extension Specialist. They usually go out. They are the boots on the ground, look for the answer for the questions that our growers raise. So we are particularly working on the diseases part. So when farm advisors or any pest control advisors find something unique and that's not known, they collect samples. They invite us to the field. We visit the field. And then with those samples, what we do is that we make isolations from the symptomatic tissue to identify what's causing it. As you can see in these the plates, we can, if we know what it look like, we identify them based on their morphology. If we don't, we go ahead and then make the DNA extraction and also identification using their molecular techniques. That techniques, if we are looking for specific the pathogen, we can screen them based on their specific primers. If we don't, we can also create the phylogenetic tree, which is a kind of their family trees, and then we identify what's causing it. Why is it important? Because if you know what you are dealing with, it is kind of like either simple to prevent mainly or manage it if we can find a way to manage it. That's all it matters. So that is take us to the research component that I'm going to share some information about that. I just want to start with some of the symptoms that we have seen in apple and pear orchards. Some of those pictures are from the mix that will give you an idea. So these are the diseases that I'm talking about, mainly canker or wound-associated diseases. For example, as you can see, this is one pruning wound. when you look from outside, you cannot see anything inside. And then when you peel the bark, you see the dark discoloration. Even though this pruning is not right pruning, which at the end of my talk, I'm going to talk to you how to do tree cut right pruning. So some of these cankers look like the European canker, but we haven't found the real European canker disease. But these are the some of the evidence that other canker pathogen can cause this kind of branch dieback. And then, yeah, right here, see, you can see like the dieback. As you can see, like the most of those cankers are started with either pruning or other diseases. For example, the fire blight um, could also be initiating an opening for these fungal pathogens to get in and then continue causing these kind of fungal canker diseases. Here's that you can see one branch that you can see those are like the dieback. You don't want to lose entire tree. Again, it causes a lot of stress. This is the one of the larger branch canker that you can see cloning. Here are some sunburn that is also made a little bit opening for those canker pathogens to get in. As you can see, they are opportunistic. As long as they find a way to get in, they just move in and then continue colonizing them. So these are the cracks, again, caused by the either sunburn or either freeze, also make opening for them. Here are a few more samples. Some of them is the small canker, some of them are bigger cankers that you can imagine. In some cases, we can also see those canker in the lower branches, which is on the trunk. As you can see here, it probably find another one. And then here's that you can see some of the secondary fungi, which is the basidiomycetes, start colonizing it as an opportunist. But you can see any canker pathogen or any soft rock pathogen could be colonizing it. So when I say the canker pathogens, they are mainly fungal species. They produce certain fruiting bodies, which we call them overwintering structures. They could be either pycnidia, which is asexual fruiting bodies. So these are how it looks on the dead part of the branches. These are the conidia or pycnidia spore that we call. And then this is the single structure embedded under the that tissue that sometimes from the outside you may not be able to see it unless you just peel it back. And then these are the spores. These spores act like a seed that can be each one of them when they find the opportunity to get into the fresh healthy plant, they can cause a new infection. Here's that I just want to show you the short video. As soon as you put a single drop of the water in that one single structure, they can ooze their spores that you can see. There are thousands of the spores, each one of them like a seed of the pathogen. Besides the asexual fruiting bodies, they can also produce sexual structures, which we call it perithecia. These structures are a little bit more different than the pycnidia because from the structures, they ooze or release their spores actively shooting in the air. So in this cartoon video, I'm just going to show you how it works. And then one single drop of the water, also any precipitation or the dew point also could activate those structures as you can see here. As soon as they get wet, they just expand physically, and then they produce a kind of tension within the structure. That tension break off, which is the structure here is called oci, and then each oci have 8 ascospores. They just release their spores in the air, and then when they find any woody tissues with the opportunistic, and then you can see the new decline in that plant. With the collaboration of Ali Andrews in the past and also other farm advisors, Collapse and Gonzalez, we did the field survey in apple and pear growing areas in California to identify what kind of fungal canker diseases that we have in California so we can identify and then find a solution. From the survey, we identified number of the different fungal structures. These are the media specific for the fungal pathogens. As you can see, each one of them are look like different, which we call it different morphology. So here's the isolation results. Most of these pathogens came up as Diplodia, which is one of the Botrytis canker pathogen, as you can see, which is the most prevalent. Followed by Eutypallata, I'm sure you have heard about it. This is the very common another canker pathogen that could go on found any woody tissues, and Diaporta and Cytospora and Falciparum. All of these are very well known the woody plant pathogens. So when we look at the species-specific identification of these fungi, we generated a multi-loci phylogenetic tree. It's no different than the family tree that, as we know, us. So they tell us what it is and then what species they are comparing with the other isomers. So as you can see, we got like the number of the species that I just mentioned. Here's the Diplodia species, which is the most prevalent diseases of the apple in California. Of course, as in the plant pathology discipline, when we identify something, that doesn't mean that is the cause of the pathogen or diseases. So in order to understand, we call it a pathogenicity test. Also, it's used to be, it's also known as co-postulates. It's the name of the guy who came up with this idea a long time ago. So what we do in this process that we artificially inoculate these fungi on healthy plant tissue to see if they are going to cause the similar symptom that we have identified. Otherwise, there are a lot of saprophytic microorganisms that could be opportunistically grow. They are not pathogen, but they could be present there. So in order to understand which one is real pathogen or not, we have to set up this kind of experiment on newly found fungi. So in this one, what we did, we just artificially inoculated in the branch, wrap it to prevent the infection from the other secondaries and also release of those symptoms as well. So when we look at those infection point, as you can see, so, oops, sorry about that. These are the infection points. As you can see, this is the control, which is just a natural dieback oxidation. But from now on, you see each fungi are showing different or length of the discoloration. Some of them are less aggressive. Some of them are really, really big, aggressive. Look at the one branch is completely dead. So that's what we wanted to understand, which one is the most prevalent and aggressive, so we can be the focus on preventing or managing those pathogens. So this is the results from the which one is showing the most aggressiveness. So these are the Diplodia seriata that shows that it's significantly different or aggressive than the other fungi that we isolated. In the past, we published this one as a first report because Diplodia bulgarica, which is the most common, that hasn't been reported in California, which seems to be one of the aggressive one. If you are interested in a little bit knowing more about it, at the end of my talk, I will show you where you can get these publications. You can access to them in my lab website. In the meantime, we also did the fungal growth difference in different temperature to see if any of those fungi are adapted on certain temperature or certain climate. You know, California is a very diverse climate from north to south, south to north, it's definitely different. As you can see in this one, that's from 0 to 40 Celsius temperature. And then, so some of those fungi are growing faster in those higher temperature than the others. So that means that they are more adapted on those areas. So this is just a general information for the scientists to better understand the biology of those fungi. So we also know that from our other research on other crops that these pathogens are primarily enter into the plant from the pruning wounds. So we have done a lot of research on how to prevent or how to protect those pruning wounds after the dormant pruning, which many of you have been doing it or have done it already, how to protect them. So one thing that I want to mention that those videos that I show you that during the winter, any precipitation, you know that they activate their spores. Presently, after any precipitation, think about that full of the spores in the air of your orchards. When you make the pruning, those can be entry point and then get in. How can you prevent it? We have done the research in two different locations with our colleagues and then found out that when you spray some of these materials, some of them are pesticide, chemical synthetic fungicide, which is the here topsin, or lunasensation, it looks like the significantly comparing with the untreated control, they protect pruning wound from those canker pathogens. This is the artificially inoculated condition. We also wanted to include a trichoderma-based beneficial, which is also known to be working on other grapevine and other crop protect the pruning wound as an organic treatment, biological, and we also found out that the trichoderma-based products could prevent the longer prevention or protection of the pruning wound than the other chemicals in our other research. So it also shows a significant comparing with the untreated control, not that great, as good as the synthetics, but that shows something. So keep in your mind that these beneficial fungi also requires a suitable condition. If your plant is stress, less vigor, too dry condition, these trichoderma-based products may not be that effective, but consistency application or year to year may increase their efficacy over time. So we are repeating that trial to find out if that's the case or not. We also tried another beneficial Aurobasidium, which is also known as a yeast protector. It doesn't work that much on this trial, but we have evidence that it also significantly works in other trial as well. Pine oil is another one that work in some of our treatments, but it looks like that when we apply on this Diplodia seriata, which is keep in your mind that's very aggressive, it is not seems to be working. As a conclusion of my canker disease pathogen, we identified a number of the fungi that are associated causing these kind of symptoms. Diplodia species seems to be most prevalent and most aggressive. In this short video that I made a long time ago to show you that not only application or protection of the pruning wound is important, but also having a right pruning is very, very important, especially for the big cuts that you are trying to make it. For example, this is an example on avocado. I used to work on this to show you how to do a tree-cut pruning method on the larger branches that I'm going to just talk to you. First of all, it's important to sterilize and clean the pruning tools, which we also know that some of those pruning tools could be transmitting those infectious plant pathogens. That's very important. What can you use? You can use either, we have a handout that I'm going to show you where you can find out those information. You can use either ethanol, which it could be difficult for you to find out, or you can use isopropanol that works. Bleach, 5 to 10%, that works very well, but that could ruin your tools, also clothes when you are applying. So that's an optional, which is the cheapest option. So you can also use the hydrogen peroxide products that could also to act like a sterilant or cleaning. So that's like the one of the tips. So let's get into the cutting. For example, we are going to cut this branch. Sorry about that. I don't know why it is happening. Okay, let's start over again. So tree cut method is that if the branch is heavy like that bigger, you have to make a first cut underneath it about like the half inch below, right? And then the second cut about 1 inch away from that. So just in case, if this branch can break off without your control heavy, this example, this is not going to cause the stripe and then a wound all the way. So your third cut should be where the branch color is, which is right there, where the plant has more the callousing abilities on the plant. Okay, so you have to make the right cut first. After that, if you can protect those pruning wounds with the products that I just talked to you, you can also find the other information about the other pruning wound protectants. That's the ideal way to protect those diseases, which is easier and organic way of prevention. So in this, the last 5 minutes of my talk, I would like to talk to you about the alternative leaf blotch that we identified in the past in Lodi area. I'm going to tell you what it is. So as you may know that most of the leaf and fruit diseases are associated with the environmental condition. In the disease triangle concept that we have in the plant pathology, you may have the pathogen, you may have the host, but if you do not have the right environmental condition, you may not have, or you may have the diseases. This is one of the case. In this orchard, we were called to identify these leaf blotch. Eventually, they turned into the fruit blotch, and then we identified. So one unique point in this the orchard was that they have been doing the overhead sprinkler irrigation, which tend to be like a rain, which take us to the northeast condition, which these pathogens are very, very prevalent. And in California, we are very lucky because we have a dry climate. We don't see that much kind of like the diseases find right condition about. But in that one particular orchard, it was created artificial suitable environmental conditions. So we identified the number of the Alternaria species, as you can see, and then some of them are more, some of them are less prevalent, and then those are the ones that was causing it. So we did the pathogenicity test, and then as you can see, we also saw the similar symptom, not on the leaves, but also fruits. They are pathogenic. But again, you may not be able to see these in very common unless you have or you create right environmental condition for these pathogens. If you are interested in knowing more about the publication that we have done so far on apple canker and leaf blotch, you can find the copy of these publications in my lab website. So feel free to scan this QR code, which is going to take you to my lab website. If you don't want to get that into that, just Google escalenlab.ucdavis.edu. You can find that. You can find those publication as well as Ali came up with a very good handout, 2 pages that gives you the very good information and then take home message. So you can also find that one in my lab website. I'm sure Ali has her own website that also put that one that you can reach that one as well. So with that, I will be happy to answer any questions. If you have questions later on, please feel free to send me an email. I will be happy to help you. I would like to thank the Ali for her help and taking the boot on the ground relationship with the growth and then help us identifying those. And also would like to thank to Muhammad Nouri, who is the former UC Corporate Extension in San Joaquin Valley. So with that, if I have time, I will be happy to answer your questions. Let me see. Ellie, do I have time?
Yes, you do. Thank you so much, Akif. That was excellent. And we do have questions in the chat and also in the Q&A. So wherever you'd like to start is great.
Okay, John Valenzuela. Okay, so you can, or percentage of the dose pruning wound disinfected that John asked. John, I'm gonna visit my lab website. You will see those concentration, but I'm just gonna mention you that full length, full strength of the isopropanol, you don't need to dilute it. You can buy it from the store. You can apply it like that. 70% alcohol is okay, but 70% isopropanol would also work in that one. But you don't want to dilute more than 70%. Okay. Every cut, it is okay. We are okay. So this is disinfection for the tools. We do not recommend application of the disinfectants on the pruning wound. They are gonna burn. There is no evidence that. For if you want to protect the pruning wounds, that's a different story. You need to apply one of those pruning wound protectants. If you would like to know more about it, send me an email. I would be happy to give you a little bit more information on that one as well. Okay,
And I
and
think we
then
have
in
time
the
for one more question. And then if you're willing to stick around, Akif, and answer more questions in the chat, I think there were some chat questions too. I
I
believe.
will.
Okay, thank you. But we do have one
Uh,
minute if you want to.
hold on. PCR testing. How? Am I done? One more question.
Oh
That's
yeah,
it.
one more question's
One
good.
more.
Yeah.
Okay. I see the chat. Okay. She said, how do you determine the culture medium? We use the common medias if we are not specifically working on the certain pathogens. For that one, we use either APDA, which is acidified potato dexter agars, or we use the potato dexter agar with the antibiotic to eliminate the fungi, the bacteria. So just to focus on the fungi. So we have those common. If you are interested in knowing what they are, send me email. I will be happy to send you that information. For the PCR testing, we do the common the primers, which is called the ITS. That will give us an idea about what kind of pathogen that we are working with. If you want to know more specific, we use the multi-loci DNA sequences of the, as I showed you, those phylogenetic tree we identify. So that requires for you to get into my lab either as a master's student or PhD student. So you will learn on that one. So that's like a good question. I think I'm going to stop right there. So Ali, just share my lab website and then feel free to send me email. Thank you, Ali.
Thank you so much. Yes, I'm going to include a lot of different links that Keith mentioned in the follow-up email as well, including that 3-cut method for pruning and how to find the branch collar and that kind of thing. That's all in that handout that Keith has. So with that, I'm going to hand it over to Jim. Welcome, Jim. We're very excited to hear from you about fire blight. So I'll hand it over. Thanks.
Sharing is not turned on, so I don't know how to do that.
Sorry about that. I think now you should be able to.
Okay. Okay, I think we're set now. Can you see my screen?
Yes, looks perfect.
Okay, so This is an update on evaluations of new bactericides, and we'll review the epidemiology of fire blight on apples and other pome fruit crops. I have to thank my crew, Helga Forrester, Luis Solari, and Doug Carey, who conduct most of these field trials. As you know, fire blight's a disease of rosaceous plants. Fire blight affects not only apples and pears, but all the groups in the Rosaceae family. It's a gram-negative bacterium called Erwinia amylavora, and as you can see on the upper left, it's a bacterium. It has a rod shaped, and it has multiple flagella surrounding the perimeter of the organism. So we have different symptoms. We know that Erwinia infects blossoms, but it can infect twigs, leaves, fruit. And once it gets into the tissues, it can go down, move into the trunk and into the collar and roots, depending on the rootstock that you have. So you can see the symptoms across the bottom: flower infection. The plant material looks blackish, as if it was burned, and so there's how the name came about, fire blight. And it also has oozing. You can see if you look closely at this, where the arrow is in the center picture, there's droplets coming out, guttation droplets from the diseased tissue, and this is loaded with bacteria. And then if it gets into the wood, it's known as a canker disease, and so it's a canker bacterium that gets in there, and it can survive as a perennial infection on the trees. So it overwinters in these cankers, and it oozes out in the spring with rain and warm temperatures. Rain splash, wind, insect dissemination are vital parts of the epidemiology of this disease, and it can survive as a epiphyte, colonizing the flower parts. And what I mean by epiphyte, meaning it's growing on the surfaces of these tissues, and it could reach high levels. 10 to the 6th is 1 million colony forming units or bacterial cells per each stigma. So this is a notorious pathogen with high populations, and it can then occur on these plants as surviving as an epiphyte. And then when the conditions are right, it can cause a disease. And once it gets into the disease, into the healthy tissue, the flower or the leaves, it can then move down into the tissues as an endophyte. So these are terms that we use in ecology of these organisms to describe how they're occurring or developing on the host plant. So an endophyte meaning it goes into the tissue, and that's where we get our cankers and necrotic tissue. One of the big differences between us and the PNW and other parts around the country is as they get their cold weather, if they don't get their cold weather, I should say, then they have what they call a rat tail bloom. And this means that, the main flowering occurs in the spring, but if they don't get enough chilling, they'll continue to bloom for weeks after the main bloom, and even go into the early parts of the summer. So these rat tail bloom, here it is on apple in the upper left here, and here it is on pears. These basically cause a lot of problems for the grower. You have to apply more protectants. You have to cut these out. So for the workers, it's extended cutting times. And again, it's the main difference between places that get chilling, where they have a uniform bloom, and places that don't get the chilling, like California. And this is one of the reasons why we have a hard time, challenging time to manage the disease. So there's different cultivar susceptibilities, just like many plant species and crops. There's differences in the varieties that we grow. I have highly susceptible for apple, the Matsu, Jonathan, Gala, Fuji appies can be devastated. There are slight variations between growing regions in their susceptibility, but there are varieties that are a little bit more tolerant, if you will, less susceptible. And these are Golden Delicious, Granny Smith, Pink Lady, Yellow Newtons. But they're basically all susceptible. And if you don't manage the disease, it can be devastating to the tree. So the way we identify Erwinia is we can take diseased tissue and we put it in water. This is just showing you the streaming of bacteria coming out of diseased tissue. And you could then take the water and streak it out and you get these colonies. This is on YDC media. And then we subculture from this to get pure cultures. We have molecular methods for identification. Back 10 or 15 years ago, there was a person that published the primers, species-specific primers for Erwinia, and we use this as part of our identification. So here we have the control in the gel, and then we have these lanes, and each lane was where we extracted DNA and ran it with the primers, and we then see if there's a reaction and separate it by the electrophoresis into, these are all positives for Erwinia, so there was 9 positives and there were 15 negatives in that. That's one of the older ways that we do it, but we also have PCR machines that can do what we call the melt curve analysis or thermal melt curve analysis. And again, you could run all your samples, and then you can characterize these primers to when they melt, and you can show this on a qPCR machine in real time. These are the positive ones that we shown up here in the gel, but this is all done on a computer screen attached to the PCR unit, and many of these are negative because there's no peak, but where the peak is positive. We then take the primers here that were positive, and we can run them into a DNA sequencing machine, and we could go through the sequencing and verify the Erwinia amylophora. So we do surveys every year with the pome fruit industry. We focus on pear and apple. These are some of the regions that we have covered in the past, Sacramento Valley and all the way into the Yuba and Lake counties. And then we also go down into more of apple regions, which are down in the San Joaquin Valley as well. We evaluate the sensitivity of the strains that we collect to streptomycin, oxytetracycline, and kasugamycin. One of the methods we do is we use the spiral plate method. This is a machine that spins the plate. Over here, the plate is spinning on this rotating disc. And then a stylet comes down, as you see in the upper right here. And we put dye into it so you could see what kind of reaction occurs. So it puts a high concentration of the toxicant in the center. And then this stylet moves out as the plate is spinning, and you get a concentration gradient of 2.5 log, with the highest in the center and the lowest at the edge of the plate. We incubate, we streak the bacteria out on this plate in radial fashion, and then we allow it to incubate for 2 days, and then we measure the inhibition zones. So here's our surveys for strep and oxytetracycline. These were done in the past. So we consistently find streptomycin resistance, and you can see different levels of resistance. We have isolates that are sensitive to streptomycin, those that are moderately resistant, and those that are highly resistant, as you see here, with greater than 70 milligrams per liter or parts per million. So these are the different types of reactions that you can observe, and you can quantify a population to their sensitivity to streptomycin. Back about 5 years ago, we started seeing people were not using strep, and the cost of kasugamycin was high, so they decided to use oxytetracycline exclusively, and some growers were putting on 8 applications of oxytetracycline, and the result was that they started selecting for oxytetracycline resistance, and we found up to 100 parts per million resistant oxytetracycline, when normally it's completely inhibitory at fractions of a part per million. So this was a new occurrence, but fortunately it was only in a few orchards where they practiced this exclusive use of oxytetracycline. In 2025, to bring you up to date, we continue to survey. We didn't find any oxytetracycline in Sacramento, Lake, Mendocino, or Yolo County, and we didn't find any kasugamycin resistance. But we do continue to find different types of resistance: the moderate resistant streptomycin and the high resistant streptomycin isolates in California. So if you're rotating between these materials, you're less likely to find resistant isolates. So just a quick review here: streptomycin resistance is divided into those that are strep A and strep B, which are plasmid-borne resistance, and then the TN5393 is the chromosome resistance. And so we have those characterized as to how they became resistant with this transposon that moved into the chromosome versus these plasmids that have resistance. And so they reflect different levels of the resistance to streptomycin. And so as you can see on the right here, we have high resistance, and this is the chromosomal RPSL gene, and that's found in California, Oregon, and possibly Michigan, where the moderate resistant one is the plasmid-borne gene, and this is shown here, and it can be integrated into the chromosome in Michigan. We found it in plasmids in California, and it's also found in different plasmids in California, Michigan, and New York. As I mentioned, 5 years ago, we saw exclusive use of oxytetracycline, and then at first we started seeing moderate resistance here. This is 0.02 at the edge of the plate, which is a fraction of a part per million, and we see that there's 40 part per million in the center of this plate. We can adjust the concentrations on the plate with the stock solution. So this was the 40 part per million, and we called that moderate resistant. And then we found in later surveys, we found these highly resistant oxytetracycline, and this is 100 part per million at the center of the plate of oxytetracycline, and the isolates are growing all the way down into the center where the highest concentration is found, and the sensitive isolates that we put on there show that it was completely inhibitory. So this is the control plate, just showing that we can get colonies of these isolates, and then when we expose them to these toxicants, whether it's strep or oxy, you can see the result of the growth curves. We tested these. We had oxytetracycline sensitive and strep sensitive, and you can see that these things are inhibiting the at 200 part per million is inhibiting the disease ability of the fire blight pathogen. But when we took the oxytetracycline high resistance, and it's linked to the streptomycin high resistance, so that means they are on the same plasmid. They're very pathogenic and competitive with the wild type isolates. So we published a paper on this. This is with collaboration with a number of people, Kwan and George Sundin and Jeju Peng and Lindsey Brown were involved, and myself and Helga Forster. So this is the plasmid. It's a novel plasmid. It's called PX11-7, which is this novel INC-X plasmid. And basically, you can see there's the strep A and B built into the plasmid, and there's the TET B gene, which is found in resistant isolates in this plasmid and is integrated into the plasmid, and it confers resistance to oxytet and streptomycin. So this is shown to be competitive, as I showed you in the previous slide, and it persisted in the orchards that we identified, but we have to realize that if you don't spray 8 times, it's still not going to be as competitive as the wild type. So in 2025, we continued our search for new bactericides. We had all these were in trials. The Coumadin has a 2L conventional as well as a new 8L formulation, which is organic. It's planned to be organic. It has to go through the registration process as a new formulation, but it is a fermentation product. Coumadin is a fermentation product, and they just took all the ingredients that were in the 2L that were not listed and put in listed organic adjuvants into the formulation. We also looked at Fireline 45 and the new formulation of oxytetracycline called NUP 17010. And then we compare it to streptomycin as firewall. Other products are the natural products, which are what's most interested for the organic growers, are these acacia extracts, which is called quam, bacterial metabolites like we've been testing with our colleagues at the University of Wisconsin, RAA, and they have an A and a B, but we tested the A for formulation. We had cinnamon aldehyde and these materials mixed with EPL, epsilon polylysine, and this is known as JAX. And so this is a new material that we're investigating with in cooperation with Summit Agro. We also looked at this Lupinus alba, Probo adverti. We've looked at Vaxiplant, the laminarin, and we've continued to work with thyme oil. As you saw with oral basidium, it's being used as a biocontrol for some diseases like what Keith was talking about, but we use it also for colonizing flowers to prevent the pathogen from getting into those things. If you have sort of like a first come, first serve, if you have the first organism there, it usually can exclude or prevent the pathogen from coming in. So that's our strategy with the oral basidium pulliens. The epsilon polylysine is, as I mentioned, it's a food additive that's regulated by the Food and Drug Administration, and it's used as a food preservative. We're the first people that were testing it as a preventative product for preventing fire blight disease. We also are testing other products like copper sulfate, pentahydrate, copper octanate, dodine, and this newer product called that's phosphite, copper, and zinc is called Curzin. It's not registered yet either, but we're kind of doing the testing to get some of these materials as alternatives to our antibiotic program. So the first thing we start out with is laboratory studies. We take the ornamental pear that's on campus here, and we put it in these flasks with water and some jib to keep them viable. And what we do is we inoculate them with a suspension of Erwinia, and then we bag them up, and then we incubate them for 2 to 3 days. And this is kind of the outcome. You see all this black flower tissue and causing disease. This is all from the fire blight. And then we compare jacks, and you see nice green peduncles. and we see nice green peduncles with the Cassumin. So we start out with the first one in A. We got high potential management with Cassumin 8L. This is the organic formulation, and we mixed it with the organic Vaxi plant, and at 12 fluid ounces and 16 fluid ounces per acre. These are the ratios that we use, and you can see high performance, especially on the first flowers out. We repeated the study you can see with different mixtures of our EPL and Cinerate, or EPL and Zacam. These are different formulations of the cinnamon aldehyde. And then we compared the JAKs at 2 different concentrations, and we compared it to Cassumin 8L. These later inoculations are also contaminated with natural occurring populations, but we inoculated it and treated the blossoms, and we can see the overall efficacy that our Cassumin 8L and our new formulations of JAKs are all significantly reducing the disease. Then in the last trial that we did, again, similar treatments, again showing that the Cassumin 8L still performing at a very high level with the new JAKs materials at different concentrations. Not as maybe as good, but significantly the same as the Cassumin with the C grouping here. So we have some very exciting organic materials for the future, the Cassumin 8L with and without Vaxi plant, and the JAKs is high to moderately effective, and we're happy to work with Summit Agro to formulate this so it'll be able to be stable and sold. Our trial is down on Granny Smith. This is down at the Kearney Ag Center, where Granny Smith is grown in that area. We compared organic bactericides to conventional premixtures like the Curzin, which is a phosphite-based material with copper and zinc added to the formulation. Our formulation last year for the JAKs, we had at low and high rates, was effective, but not the best treatments, as we see with the Cassumin 8L separating into the highest level of performance compared to the control. So we were getting about 7.5%, and with the Cassumin 8L down to 1% incidence of blossom infection. And with the other treatments, intermediate, still showing significant reduction from the untreated control, the UTC. This was on Granny Smith. We did the similar trial on Fuji apples, and again, similar types of results. We had the Cassumin 8L at the highest level of performance. Instill ProBlad, which is organic treatment. Blossom Protect with the buffer and the adjuvant. These things were still a degrouping and showing significant reduction from the control, the UTC. Curzin was intermediate in its performance, and Alum was intermediate in performance in our trials. The same with the Cassumin 8L mixed with other materials like Fireline or Silit. But the Cassumin Vaxi plant did pretty well. It's still in the D grouping, so we can't really say that it's different, but it's working. Again, we're under higher disease pressure in this particular trial. So we were working at Davis on Shinko apple pear, which is the Asian pear. It's now called apple pear. Again, we were looking at the performance of our JAKs and cures, and we got significant reductions. You can see the low rate of JAKs is not working as well as the high rate of JAKs. This is the Blossom Protect, it's intermediate. But then we have the Cassumin 8L with Vaxi plant, again performing very well, similar to the oxytetracycline mixed with Silit and Instill ProBlad, all in the D grouping. So these are, again, summarizing moderate effectiveness of some materials. We're testing different formulations of JAKs, and we're testing organic formulations of Cassumin. And the NUP is a new formulation that's a 45 WG and we hope to have that soon available from the registrant for oxytetracycline. So to summarize here, our treatments for managing fire blight, we have direct toxicants. All these materials on this side are usually highly effective, high performance, but we have a high potential for resistance. And that resistance is shown with every year in our trials evaluating isolates to the streptomycin. We are lucky that the oxytetracycline is only found in limited orchards, and we haven't found it widespread. And the Coumadin is still very effective in our experience in California. We mix it with Silit, which is similar to Dodine. These are things that bind to the membranes of these bacteria, and they help toxicants like copper or these antibiotics to get into the plant. As I mentioned, the phosphonates do have some suppression effect on the fire blight pathogen, and where other things like the biologicals are competing and suppressing the pathogen. And so some of our new things are to look at these materials like Sacan and Incinerate in mixtures with the EPL, the epsilon polylysine, and that's where we get JAKs. We continue to test QAM and RAA as potential registrants. RAA is going through the EPA now, and QAM or QAM is the material being offered now by New Farm. So the summary is we have direct toxicants. We're testing new products like Coumadin AL and Curazin, and we're testing natural products like RAA. We hope that these can be listed as organic, the Coumadin AL and the RAA, and the Curazin, because it has phosphite, will not be. We then have the JAKs, which is this EPL cinnamon aldehyde mixture. The QAM is the acacia extract, and Selectricide we're testing. We did test in the previous year, but it was we couldn't get a hold of the material for this past year, but we're going to be testing it again this year. So we have biocontrols, systemic acquired resistance materials like Actigard and Apogee. We also have shoot growth inhibitors like Apogee. And the key is to have an integrated management program, and pruning and timing of the chemical treatments are very essential to protect the susceptible tissue. This is summarized in our fungicide, bactericide, biocontrols, and natural products document posted on the UCCE website. And you can see this as myself, Michaelides, and Akif put this together. And there's all the rankings of the conventional bactericides as well as the bactericides and biocontrols and natural products. And we use a 0 to 5 scale to characterize their effectiveness. So obviously we need to think about the rootstock signs, the orchard location, the design, which determines the air movement, the avoiding high fertilization, avoiding over irrigation, which are all conditions that favor disease. Sanitation is important, removing cankers, removing infected tissue in season, and removing alternate hosts in the Rosaceae family from around the orchard. And cleaning and pruning, as Akif mentioned, with sanitizers like bleach or PAA or these other types of things. And chemicals are also being used. Obviously, we're trying to get an antibiotic, the Coumadin AL, as organic in the future. And we use early season copper, we use biologicals, and we rotate between these different products. And as like we have Apogee, we try to suppress the new shoot growth, and we have rotation of different modes of action as our mechanism to prevent selection of resistance. I'll stop there. Thank you very much. I just kind of went over a little bit, so I'll let Elle take over now.
Okay, thank you so much, Jim. I really appreciate it. I saw we had just a couple questions in the Q&A and maybe the chat as well. So if you don't mind sticking around another minute or two just to answer those in the Q&A, that would be great. But we're going to go ahead and switch over to Jalendra Rajal's presentation. So welcome, Jalendra. Thank you so much for joining us to talk to us about codling moth. I believe I've made you a co-host, and so I think you should be able to share your screen when you're ready.
Yeah. Good to see you. Good to see you, everyone. Good to see you, Jim. Okay. Let's see. Can you see it? Um,
We're seeing multiple different slides right now.
okay, continue
If
to
you
see.
go into presenter mode, I think that should work. You go
And
back.
maybe that's the, okay. Is that
Yes,
work?
perfect. Looks great.
Okay, great. The two screen thing. Okay. Sounds good. Yeah, good afternoon, everyone. My name is Jalendra Rijal. I work with the University of California Cooperative Extension in Modesto. I was trying to get in Modesto, but I cover northern San Joaquin Valley. So I'm entomologist. So whatever I do is related to the insect and insect pest management. So for today, I know that we talked about a little bit of organic management, but I think it's in general for managing any insect, whether it's organic or conventional, we apply the principle called IPM. And so I'll talk a little bit broadly about this insect IPM tools and plus also some of the particular tools that is applicable for organic production system. Okay, with that in background, so talk a little bit about a cardling moth. I think it's most of the time, I think insect identification is, or it understanding its biology is, it's important. It's critically important for managing pest. So this cardling moth, you can see it's a little tiny moth, about half inch. You can see the coloration there. They have this kind of coppery structure on the sort of back of the wing. Kind of posterior side of the wings. You can see, and these are some of the cardling moth that is captured in the traps, so in the sticky traps. So you get idea about how this look like. They look like if you ever use the cardling moth lure and traps, then you'll certainly capture these in the traps also. So there are destructive pests, of course, in apple, and it has a multiple generations, and also it's a key pest in walnuts too. So the apple, pear, and walnuts, those are the ones that we deal with in routinely, at least in California, and of course in throughout the United States in terms of the apple and and also in pear. So other host crop include crab, apple, quince. You can see some of these, sometimes occasionally in peach, but they are not as severe compared to the other three major ones I talked about. Important thing to note is even though it's a moth, so they lay eggs, so the larva is the one that caused the damage, right? So most of the time, moth would feed on something liquid or nectar or something of that kind and live for a couple of days or maybe week. But most importantly, the larva is the one that actually caused the damage. So these are some of the example: apple, carding moth larval feeding on an apple. You can see in the pear here, kind of like a tiny holes in here. And then this is a cardling moth in walnut. So kind of more or less same principle, right? So the lay eggs on somewhere in the fruit or leaves, the larva will get into the fruit and cause damage that way. Okay, so if you look at overall kind of idea about cardling moth is pretty much ubiquitous throughout the world. It's a pretty much global pest for majority of these tree fruit crops. And here you can see the distribution, of course, United States and some of the Canada, where the apple is grown. It's commonly found, so they have this kind of light shaded is a carding moth host is present. Carding moth is present and as well as the host. So these are the area where we see that damage, of course, and here is a bunch of places in the Europe, also North America, South America. So pretty much all continents that has carding moth activity. There are some the area where there might not be present as a record, but we suspect that there is a codling moth already there. So this is indicated by this. So in general, I mean, I don't need to go in detail into this, but it's definitely a global pest of that significance. So okay, so that's a little bit of background of codling moth. So codling moth, and late in the season, they're later stage of the larva. They overwinter in the plant. So in the plants, basically in the barks where there is found the rough surface or places where they can hide on. So they basically overwinter in there. And then so after the winter, of course, in March, kind of late March, early May, that's where we're seeing, well, not early May, late March or early April, that's where we're seeing the carding moth activity begin in the valley. And it's more or less similar in the coastal area too. Again, it depends on the temperature. You know, there could be 7 days, 10 days, 2 weeks differences depending on where you are. Warmer the temperature, you likely see their activity begins in earlier. Right? So female of these insect, the lay eggs, you know, overwinter female. So that means that the female that is the they overwinter as a larva, they pupate there and the adult emerge. So those adults will basically lay a little less number of the eggs compared to the seasonal, which can lay up to double after overwintering. Female can lay eggs. And they can lay eggs on leaves. Mostly it's a peaches and apples. You can see the eggs on the leaf also, and then and of course on the fruit also. In walnuts, mostly it's on the fruit, but they can lay eggs on the leaves too. They have a 2 to 4 generation. So depending on the where you are, right? I mean, it's also the global past. So some other places that might have less or more generations, but at least in the valley, in the Central Valley where I'm based at, we routinely see the 3 generations per year. So there could be some coastal area, there might be 2.5 generations, depending upon the temperature, but anywhere from 2 to 4 generations. Larva of the all generations, basically in apple, talking about apple, can be economically important, right? So whenever the apple fruit is there, when they get a chance to lay eggs, all these generations, the larva can feed on and cause damage. If you looked at the overall life cycle, again, I already described, but maybe it will help you to kind of remember a little bit better. So these are the places where larvae overwinter, they pupate in there, and then the adult emerge in the spring, the beginning in the spring. They mate and then lay eggs on the leaf or the fruit. After that, the larvae get into the fruit, they feed on it, complete their multiple stages of the larva. Again, they come out and pupate somewhere on the similar way, and then that continues, right? So and then the next generation will again come back and lay eggs and continues like that. Okay, so we kind of understand it's a moth pest. The larva is the one that is causing the damage. The lay eggs on the fruit and on the leaves, and then those tiny larvae will get into the fruit, and that's how they do the damage. So how you manage it, right? So understanding that the whole thing that I describe about the biology of this insect is important, right, for making IPM decisions. So what is integrated pest management in that regard? So I want to say the integrated pest management is the concept that can apply to manage the pest in conventional, organic, any other kinds of sustainable system. You apply these principle or a strategy, right? So that can fit into the all things. it's really a decision-making process with the, coordinated use of multiple tactics to optimize the control for all class of insect, if you think about it. But here we're talking about insect, we are talking about carding moth, um, in a manner that ecologically and economically sound. So that's kind of broad definition of that. What that means? That means is that it is important to identify what pest you're dealing with because that is associated with its biology, behavior, and that's important to understand first. How do you track this insect? How do you track these different activities throughout the season? So monitoring of that insect is important because you can't control the insect that are in all different stages, right? So there should be the targeted stage where you can control that insect in order to find out what is happening in the field. Then you need to have some sort of criteria, and in this case that we use the monitoring tools for this insect. Sometimes we use the threshold, right? So not all the times the threshold is available, but when it's available, then we can use the threshold. The idea is that, if you're catching a 10 moth a night in apple orchard, so that is definitely high pressure orchard. So you need to think about how to manage that, right? Just the criteria and use the various preventative as well as curative techniques to manage this pest. So altogether, if you think about knowing the insect, how you can monitor it, if there is threshold, apply that to make a decisions. And once it comes down to decisions, what are the tools available that you can effectively use to manage this insect? And the goal of the whole IPM program is really ultimately in a conventional as well as organic, the same way. It's the conventional system, there are different types of insecticide you use. Organic, you use the different kinds of insecticide, but the whole principle is the same. Is for IPM is to reduce the use of pesticide that are harmful to the environment, to the people, and then also it has to be done in a way that it should be economically it should make sense economically, right? So if you're not able to produce it economically, that's not sustainable production of your crop. If you're doing it for that purpose, if you're doing it for backyard for your own consumption for other the ways, then economics might not be a big factor. But in terms of overall IPM for applying to commercial production, whether it's conventional as well organic, economics is important. Okay, so I already a little bit described how it works in terms of how the codling moth will cause the damage. Here are some of the picture. You can see that they're egg laying on the leaf. You can see the very sort of like a oval and flat eggs. It's very difficult to see, and especially when they are cream white color, it's hard to see them. But the tiny larvae will come out from there, whether they lay eggs on the leaf or the fruit. And from that point on, they start penetrating inside the fruit. And so some of the majority of the cases, the larvae start penetrating in the fruit from that stem end right here. And so that they can go in and cause damage. So you can see a lot of example of that and making holes and different stages of the larva potentially make different holes. So and then to the fruit at either a stem side or the calyx side, right from the top, because these are the places where they can securely go and dig into and then enter or into the inside. Damage symptoms, of course, you can see, it varies, right? So if you have the tiny larva getting in, you might not see even any of that in there. But whenever they come out after those feeding inside, and when they come out, that's where you can see these big holes. And so those are the symptom in there. And then with the frass and everything else there. Okay, so now pest monitoring, right? So that we talked about the monitoring is important. Why is important? Because if you think about the whole life cycle, you wanted to apply any type of insecticide or control methods, at least the particular insecticide, targeting that early egg stage and or that early larval stage, right? So in order to find out, you need to have some sort of criteria to know when that is happening in this multi-generation insect. Um, so in order to know that, we something called the bifix. That means that they're overwintering in the winter time. They pupate there, and then in spring they emerge, as I mentioned earlier. So whenever the emergence happens, when you see that consistent emergence of the adults, that shows the biological activity of that insect is begin for the season, right? And so that is what we set as the biofix. And there are different insect has different way to set biofix. Some insect use January 1, and then you can accumulate degree days from that point on. But for cardling moth, we use these traps, pheromone traps out there. And whenever you start catching the pheromone moth in those pheromone trap in a consistent manner in a week after week, so that first week when you start catching is the biofix for the codling moth. Also, one caveat here is that also the evening sunset temperature should be more than 62 degrees Fahrenheit. So if you apply this criteria, you can set the biofix, which is like a date. Let's say if March 28 is the biofix for this insect in 2025, right? So that's your date. So now you want to apply the degree days concept, which is again, this is the days, but it's measurement of the heat units instead of calendar days, right? So it is calculated based on the daily maximum and minimum temperature. You have a weather station in many places, so where you can calculate the maximum and minimum temperature for those um, um, stations, and you should be able to calculate how many degree days you accumulate from that biofix as you move on. And again, every insect has their different criteria, and based on that criteria, you accumulate those degree days. Why is important? Because the temperature is directly related to their growth, development, reproduction in insect world. So if you see the increased temperature, the activity also increase like that. So, there could be the day with, like, let's say 75 degree with the degree maximum and 60 degree minimum temperature that day. And then the next day, if it's 65 and 60 degree, that is 2 days. But for the insect, the second day is less prolific, right, than the first day. So that's why we wanted to use these heat units as a criteria to measure how much they develop instead of calendar days. Hopefully, I make it clear. Okay. So for the trapping, in order to set biofix, I talked about, we can put the traps out and then set the biofix. So there are multiple traps available, types of the lures or attracting available for this insect, carding moth. Generally, the pheromone lure, 1X lure also we called. This is what you can put it into these traps and place them in the orchard. So these traps, these lures have the male pheromone, these have the female pheromone that attracts males. So that's the criteria we use it. But sometime when you have mating disruption and other in that scenario, which I'll talk a little bit later, in that case, you will have your male trap would not catch any males because the mating disruption disrupt their behavior to finding the traps. So in that case, you use something different kinds of trap. It's called CMDA combo lure. Sometimes there is another one, CMDA. Well, there is one called CMDA. There is another one called CMDA AA with acetic acid in it. So these are all commercially available. So if you have a questions about this, contact your local farm advisor, pomology advisor, or IPM advisor. They can help you in figuring out. But we have a commercial lure available for carding moth to use in any system. So once you have that biofix you have, and now you can calculate the degree days using this UC IPM degree day model. If you have a hard time to find out, I would suggest that Google UC IPM degree days. If you go to that page, you can see the list of the insect in there, and you can click the carding moth, and it give you the different idea about their temperature and things. But most importantly, your county, and then when you select the county, and it also give you the weather station where you can select your weather station and set your biofix, and you just boom, you calculate the degree days like this, right? So let's say biofix was April 8. From that point on, you accumulate these biofix date, and it goes on like that. And why it's important. So for example, if the activity begin here, 250 or 200 degree day is the when you should target it if you're targeting the early stage of the larva, right? So if you use this trap as well as the degree day biofix date, you should be able to pinpoint when that 200 or 250 degree day will hit. And so that would be your timing to treat for that particular generation. So this is just an example of that. So this is from walnut, but it should not be different. Too much different. So this is the first flight. You can kind of look at these. Blue color one is the moth flight, and then second flight and third flight like that. And these are the degree days. So we have a 3 degree day, 3 generations of the carding moth, and that degree day timing from that particular locations, weather station, help you to set what is the timing. For example, spray timing 300 degree days, June 26 for this flight, second flight. For third flight, that's August 9, right? So that's how we're trying to get out of these methods. Okay, so also for carding moth, you can also use the fruit sampling as a criteria to see how much damage it occurs and that kind of thing. So there are some of the criteria you can follow. UC statewide IPM guidelines for apple, and it should give you the idea about how many fruit you can take and look at how much damage it has and that kind of criteria. I won't have time to go through in detail here, but that's another way to know how much damage historically in your orchard, and then you can also compare last year versus this year versus next year, so this will give you a better idea. Okay, let's talk about some of the IPM tools. So for the IPM, there are insecticide tool, biological mating disruption, some cultural mechanical tools, so we'll talk a little bit more about those. One of the thing, I mean, it depends on your production practice, right? So if you have a couple of trees or you have a small acreage and things, you should be able to apply some of this. One of the thing is that because they have a multiple generations, so when the fruit when there is damage happens on the fruits drop. And so if you have a lot of fruits, so they're abandoned fruits, so cleaning and sanitizing and always helps to reduce the population. Also, they overwinter in those barks and other things. So other things you should be able to do it is use this cardboard type and then put it into the branches or mostly in the trunk. And that's the larva thinks that's a good place for the overwintering. They will just come there overwinter, you basically remove and kill them, right? So that's another way of doing it. Fruit bagging, fruit netting, tree netting, these are some of the tools and practices people use it. Again, it depends on your production system. I mean, it may not be feasible for 20,000 and then every single fruit you can bag them, right? So these are some of the cultural or mechanical practices that might fit into the production. Another most important thing for the commercial production is mating disruption. So there are mating disruption products that are fit for organic production system. So the concept of mating disruption is using the sex pheromone that is chemically synthesized in the lab, and that used to interfere the mating of this insect. This is a really good thing because you're not talking about the chemical insecticide in this case. You're talking of some of the pheromone to do the behavioral control of the insect. And all of these insect has a very robust mating disruption practices, and it has been used widely throughout the United States, depending on what crop or pest you were talking about. And again, this is a very selective, residue-free, very well compatible with the IPM system. So what is mating disruption? So mating disruption, when you have no mating disruption, what happens is that female produce pheromone, and that plume, the male moth will follow that plume, and mating occurs, and then reproduction happens, right? And in the mating disruption scenario, you put the mating disruption with the different methods, and you have thousands of plume instead of one. And then the male moth will essentially need to find the one single plume that is coming out from female. So in that case, the male become confused and would not be able to find the female, no reproduction. There are different mechanisms behind it. I probably won't have time to go through, but at least for codling moth case, what happens is that when you put these massive amount of pheromone out there, there are thousands of plume, and that this male has to actually find that original plume from female. So if you have thousands of plume there, essentially the male, he has to compete. The female produced pheromone has to compete with all these artificial plumes, and then male basically most of the time following the false trail, and so it just continuously not finding the female. So it depletes its energy, and then and would not be able to find a female. Or in other words, it can delay mating. You can find a female, but after 3 days, 4 days, which is long for insect life, right? So if that's the case, also they will be able to reproduce, but it's small amount of eggs, and then the eggs might not be well fertilized, and those things. So it basically population reduction approach. There are multiple options for it. So you have a sprayable pheromone. So people target it for this different flight because you know that you have adult activity of the different flight. You know the timing, apply around that time. It is, it's a decent, but it's not the most preferred way. There are other options where you can use the plastic dispenser, hand applied, as you see in the pictures. There are some different company producing it in different rates. Also, there are aerosol dispenser, which is like this, where this canister has the pheromone. They puff the pheromone in every 15 minutes or so, and that's how the pheromone disperse within the orchard. For codling moth, more source point of the pheromone is better because of the competitive nature of that. Because they need to compete with the female produced pheromone, and so preferably, more units is better, but at the same time, you might think about a little bit more cost for those. Okay. So other natural enemies, biological control aspect, there are a lot of predators and things out there, but in the commercial production system, I don't know how much all of these will help you to completely control, but it always helps to reduce the population. There are some entomopathogenic fungi and virus and other things that are commercially available too. So which I'll talk next. So on the biological control side of things, one of the things that is commercially available is called Trichogramma platneri. That's a species of the Trichogramma. That's a species. It's a lay eggs on the eggs of the of codling moth, and it's turned into the dark color. So instead of the codling moth larva coming out, basically these wasp larvae will survive in them, right? So they kill these eggs or feed on it. Um, but there are, it might fit or might not fit. There are some trials shows that there are reduction certain level, but not as complete control. But it can be something that you can be think about. Other consideration to think about, how you can, if you release them, how you can protect them from the broad spectrum insecticide, or how you can protect them, or how you can use some of the plants like insectary flower and other nectar for that wasp, trichogramma, to survive there and continuously provide the service to your orchard. So those are some of the considerations you need to think about. Another important one, commercially, a lot of product available is the virus. So it's an insect pathogenic virus. So it's caused disease to the insect. And so these are some of the products out there, commonly used products, multiple variants, isolates there. After using a multiple year, it can get again resistant, but the company are good to know that, and then they will switch the product and variant and isolates and things of that kind. One thing to keep in mind is that when you apply them, you need to apply coverage. It's really, really, really important because insect has to ingest the product in order to be effective, right? So also when UV lights and other things, the virus are sensitive to that. So those kind of things, they have short residuals. So you might think about using multiple times, rotate with the other insecticide like spinosad, or use the mating disruption and use some of these additional tool like that. It also suggested that avoid some of the copper fungicide, lime sulfur, BT product, just tank mix, avoid tank mix of those. So other insecticide options in a conventional system, it's pretty common. There are some of the diamide, and some of the newer type of insecticide like Spearlap, some of the growth regulator type insecticide also available for commercial production. For the organic, the most common one is Intrust spinosad, somewhat well, that also can help the carding moth eggs. Killing the carding moth helps because of the egg, because of the smothering effect. Surround is also pretty effective. And as you see in here, there is a steroid insect technique available, but I don't know. It's for the small production. It's not that effective. But in Canada, they are using it as an area weighted approach. So hopefully, in some point in the future, in pear growing areas or some other areas, we'll be able to see that. So I know I ran out of time on 31 minutes. So it's an important past you understand its biology, and then use these tools that you can predict their timing, and that's how you should be able to manage this insect with the available tools that are out there, including mating disruption. So with that, I would like to thank everyone, and hopefully we'll be able to answer some of the question. Maybe in a chat.
Thank you so much, Shalendra. That was great. Yes, let's take maybe just 2 or 3 minutes to answer questions live. If you want to look through and just answer any questions, that'd be great. And then we're going to take about a 10-minute break after that.
Okay. Sounds great. Okay, so I can go live a little bit?
Yes, go for it.
Let me see. If you're trying to collect the carding moth in the cardboard around the tree, when do you apply the cardboard, and when do you dispose of it? Yeah, should it be disposed in the garbage, not city compost? Well, it depends on the when you are targeting, right? So if you are targeting for like overwintering population, then you should do it in the fall when the codling moth larvae are still active on the fruit. They have not overwintered. They are not moving into overwintering. So you apply in the fall, and then that goes throughout. But that's a long time. But if you want to do it, and it has a multiple generation, so you could also do it is in the middle of the summer where there is one generation coming and then another generation starts. So around that time. But how do you know about it? It's you need to track the trapping, you need to track the adult activity as well as use the degree day, and that should give you ballpark timing, right? So it's not 100% exact, ballpark timing when you should be able to do it. But for the overwintering, definitely in the fall. Okay, so are firm rings effective in repelling male moth? I don't think so, because the pheromone that used in mating disruption is not exactly the same pheromone blend used in the by the female, right? So in other words, when you have um, so yeah, in general, so when you're talking about the mating disruption, we're not talking about the exactly same pheromone that is produced by the female. It's close enough. So in that case, you're not repelling them. But if you put the traps out with the female pheromone in it, then because that trap has the pure female pheromone, the male moth would not be able to find that trap if under mating disruption. So that's the one that you need to think about. While fruit is on the tree, can I spray the props with something? Any suggestion? I cannot remove the wooden props while fruit is on the tree. Um, can I spray the prop with something? I mean, I think um it's I would still suggest that the removing would be probably the best way. If you spray, um it's still you might not get the 100% inside the cardboard. Um, so that is one of the things that you need to think about. If you're able to do it effectively, then you should be able to do it. But if you have organic system also, I mean, their insecticides are not like also 100% effective. Or I should not say that, but you may not get the 100% control of that. And so I would still think that, it's still best way it would be to remove the props and destroy them. Traps, though, traps were only used to figure out biofix data. Okay, does mating disruption mean traps? No. So as I mentioned, the mating disruption has a synthetic pheromone alike, right? So it may not be the exact same blend what female produce, but it's close enough that it disrupt the male behavior. Um, and used to figure out biofix. Yeah, essentially, in the very beginning, when you use the traps, that you're using it to set the biofix. But also, if you're continuously using it, then it gives you those moth counts where you can see the activity going and then coming down. And once the activity coming down, that's where you need to think about setting another generation biofix. So the traps helps you on that, because if you are zero or very low number, that means that there are ending of that first generation. Now, any point the second generation can begin. So whenever you start seeing the increased number of the trap counts from that deep, that should be your second generation. So traps should put throughout the season and should help that way.
Okay, thank you so much. I think just in the interest of time, because it's 2:40, I want to give folks a break. So we're going to take a quick 5-minute break. Can get up, stretch, get some water, and then we'll be back at 2:45 to learn about cover crops. And if there are more questions, feel free to put them in the chat or the Q&A. And Jilendra, if you don't mind sticking around for a minute and answering them, that would be awesome.
Yeah, we'll do that. Thank you.
Thank you so much. Okay, we'll see everyone in 5 minutes. Okay, welcome back, everyone. We're going to go ahead and get started soon. Next, we have Kamyar Aram talking about cover crops. And yes, that looks perfect, Kamyar. Thanks. Whenever you're ready,
Okay,
take it away.
great. And I hope you can hear me well too.
Yes, sounds good.
Great. Good afternoon, everyone. And yeah, thank you, Ellie, for the invitation and great webinar. So my name is Kamyar Aram. I'm Specialty Crops Advisor, similar role to Ellie, actually, down in Alameda and Contra Costa counties. And in my program for the last 6 years or so, I've been learning about and experimenting with cover crops. And so 6 years, I still feel very much that I come to you as a student with some experience. But cover crops are a big topic. And so just to get a sense, I have a lot of material here for pacing myself and for emphasis. It would be great if you want to share in the chat what you feel like your experience level is with cover crops. If you're just hearing about them, like you're completely new to them, or you're a beginner, maybe you've tried them out or just learned a little bit about them, or intermediate to advanced if you've already been practicing. So if you want to throw that into the chat, that will help me kind of pace myself and emphasize different things. Cool. So, one of the important things to say is that, again, that cover crops are a very big topic, and I feel like no one presentation or article or even book is going to give you all the information to detail all the intricacies and all the possibilities. I was trying to think of an analogy, and I think it's perhaps something maybe like cooking might be a good analogy. But so for that reason, I want to put, this is an overview of what I'm going to try to cover today in the next 30 minutes or so. And so I'm going to put the resources first, and we'll go over those, and then cover the concepts and principles, the benefits and concerns, the different types of cover crops, overview of practices, and then finally take a look at how to manage some of those concerns. So here we go. First, there are some really great kind of handbook size publications from UCANR and UCANR and partners, including cover crop best management practices for almonds, the cover crops for walnuts and for vineyards. None of these are, of course, directly addressing apples or even fruit trees exactly, but a lot of what's in these books will be cross-applicable, and we'll kind of touch on some of the nuances. The Interesting. I thought, oh, okay, here we go. So UC which is a Sustainable Agriculture Research and Education Program, has a great website called California Cover Crops Resources, which includes grower profiles, among many other things. A really nice kind of a resource you can explore. The grower profiles are something I really appreciate. It gives you kind of individual experiences and experiments with cover cropping. And back some years ago now, my program, along with our Contra Costa Resource Conservation District, did a collaborative effort, actually with our urban agriculture program too. Then, and we produced a series of videos. We called these the Cover Cropping Opportunities in Specialty Crops series. You can find that at the link. And actually, one thing I forgot to do was going to do it at the beginning is I'm going to try this out. Hopefully it works. I'm going to just drop all these things into the chat, links to these, and hopefully you can copy them and access them in time. Let's see, did that work? Oh, that's not going to work like that. Let's see, to everyone. And, there we go. Hopefully you can access that. So, So that the link for this one's there too. But yeah, cover cropping opportunities and specialty crops, really some great videos featuring a lot of renowned, at least in the world of cover crops, growers and other experts. Community Alliance with Family Farmers is a group that I've collaborated with quite a bit, not in cover crops, interestingly, though, but they have some really wonderful resources, really brief and to the point and comprehensive. So I do recommend looking at their stuff. And then the Western Cover Crop Council has a really nice website of resources. So that's an overview. There are many, many more, of course, but those should be great places if you're not familiar with them to get started or to deepen your understanding. So moving now, just asking kind of a quick review of what we are talking about when we talk about cover crops. These are annual field crops for the most part, and often really are the same species used as forage and sometimes as grains and legume seed crops. And they are planted in orchards in perennial systems and vineyards. They're planted in the middles or the alleys in order to protect and improve the soil. And they can have many other benefits we'll discuss. Generally speaking, they will produce more biomass than the resident vegetation, or the volunteer vegetation. And then another important thing is that the benefits accrue over many years. This is not a short-term kind of solution or practice. So why practice cover cropping, or why implement cover crops? The number one reason is to cultivate a healthy and well-functioning soil over time. And often another reason that is listed is to address specific management concerns or goals. These can include like water infiltration, nitrogen management, pollinator forage. But except for maybe the pollinator forage, most of the benefits of cover crops are really like water infiltration or nutrient management. These really relate to healthy or well-functioning soil. And none of them, again, this is kind of like adopting healthy eating or exercise habits. It's not something that's going to, you might see benefits from this 1 year of practice, but they will be short-lived and will be limited unless you integrate them into a regular kind of part of your farming practice. So there are generally different practices of cover crops that can be divided into the winter cover crop, the summer cover crop, and then perennial reseeding cover crops. For the most part, I emphasize winter cover cropping. That's mostly what we do in California, but in orchards and vineyards especially. And this is because they grow during the rainy season and require no irrigation. And water is generally a precious resource in California. So we're not, and when we're watering a cash crop, we're not looking for competition. So these are grown when the trees are dormant generally. So we're talking, of course, about mostly in deciduous species orchards. And then they're terminated, or, their growth is ended in the spring. However, in apple orchards as well as other fruit trees, there might be some suitability actually to thinking about perennial and reseeding cover crops, or even summer cover crops that grow during the growing season. Especially if you have U-pick operations, or anywhere where people are walking around and you like to have a kind of almost like a lawn or something, where people or equipment can kind of just have a nice ground cover during the summer. And those tend to be the summer cover crops. If you're going to practice them in orchards, tend to be grasses and clover, like legumes that are low growing and produce less biomass, and therefore going to have somewhat less of those soil improving benefits. So mostly I'm emphasizing winter cover crops. Now, this is a picture, after this, this was a multi-species cover crop that was mowed, and the grasses grew back. and they kind of just persisted. This is a sprinkler irrigated orchard, so they grew back and were mowed, and they kind of became a sort of summer ground cover. Although you can see, because of the way they were established, they're a little bit thinner than you would typically have for something that you wanted ground cover for. In any case, let's get to looking at the cover crop benefits versus some concerns. The main benefit, of course, as I've mentioned already, is soil health, which I think of as kind of synonymously with soil function. This includes things like water infiltration and nutrient cycling, as well as biological function and diversity. Over the time, I used to always call the uh, early access, but so I'm calling this sooner post-wet entry. I hope that doesn't sound, I'm just curious if that makes any sense to you. This is basically when you have rain or irrigation, you can get into the orchard quicker than you would if you had a bare ground. And then weed suppression can be another benefit. And then I put carbon sequestration in parentheses because that's not really an agronomic goal generally, but because we have some incentive programs, it is can be a conservation or ecological goal. Some concerns, of course, in California is water use, frost risk. I'll discuss. There's some worry that cover crops may enhance frost risk in the spring during bloom. That's a big deal, of course, especially with something like apples, depending on what kind of variety they are and when they bloom. Cost, of course, there is some cost to the practice. Whether you can get winter orchard activities done, and then whether they might enhance pest activities like gophers, nematodes, or even other arthropods. Looking first at soil health, I mean, we're really talking about adding organic matter to the soil. This improves physical function, structure, water infiltration and retention, protection against erosion. Biological function includes, really we're feeding the microbes in the soil and some of the invertebrates, not micro rather, but some of the small invertebrates. Improved structure, aeration based on biological activity, and then even disease suppression, possibly. And then for water infiltration, of course, the roots of cover crops open channels in the soil. And all cover crops will do this to some extent, but these mustards are typically really strong, have big tap roots, and kind of dramatically demonstrate this. Organic matter improves soil aggregation or pore structure. And so you may have seen these tests before where you take a clump of soil and you put it on a mesh in a cup of water and see how quickly it falls apart. And so you can see organic matter helps soil aggregate and hold its structure better. It also just directly can increase the water holding capacity of the soil to some extent. And then vegetative cover and organic matter both prevent crusting, and so allow, prevent water just from running off when it comes in high quantities. Nutrient management. So legume cover crops can contribute nitrogen. We'll talk about that more through their nitrogen fixation. But all cover crops, especially grasses and even the mustards or the brassica, will forage nitrogen and hold it, so keep it from leaching out of the soil during the winter when it's raining. And then when you incorporate them back in, they will release that back. And also organic matter improves nutrient cycling and nutrient holding capacity of soil. Okay, so early post-wet access, if that's like a reasonable term, but yeah, basically when you have rains or when you're irrigating, if you have a bare soil, it becomes muddy. You can't get out there with equipment, and even sometimes can't really get out there in person on feet without getting a ton of soil stuck to you, if depending on the soil type. So cover crops take up water and they provide attractive surface. This is really good. Just in this example here, for example, for early spring planting of a new orchard, this was a grower that planted a really nice dense grass and was able to get the equipment out there to plant. And some cover crops will not survive trampling of equipment, although grasses tend to pretty well. And then weed suppression is definitely effective. I mean, you can see from this this stand, like very few weeds grew in that kind of stand, very competitive grass growth here. But if you don't have as well of a establishment, it may not work as well. And then the non-planted area, your tree rows, when I say non-planted, not planted to cover crops. So your tree row is generally kept bare, even with cover crops, and that will require weed control, whether it's mechanical or chemical, depending on your practices. And then cover crops can function as a mulch if not incorporated and suppress weeds that way. I'm pretty sure this image shows what is called a the name is now escaping me, a roller crimper tool, which basically just knocks the cover crop down and pinches it so that it functionally dies, but doesn't really chop it up at all. So here's some images from a mid-season in a trial that I had. You can see the bare soil, a little bit of mulch left from a very large biomass producing mix. And then in the far right, that's all bindweed. So, these mulches can suppress bindweed and other weeds to some extent. Of course, not completely, but they do function that way pretty well. And for carbon sequestration, this is a state priority, and there are incentive programs that can help you offset some of the costs of cover cropping. So that's really nice because there are programs like the Healthy Soils, California Healthy Soils, or USDA EQIP grants that can, cover the cost of cover cropping as a means for better carbon sequestration for the state, but also you can get all the benefits for your orchard. So, talk with your NRCS or Resource Conservation District rep to see if that's something available to you. Also, there are incentive programs from Seeds for Bees for pollinator support. So check them out to projectapsm.org. That's a nice program too for cover crops incentives. So, okay, let's quickly go through the different types of cover crops. And I alluded to some of these already. There are, of course, the main groups of cover crops discussed generally, you'll see in guides are grasses, legumes, and mustards, or the brassica species, which of course are also forage species, as I mentioned. They're not the only ones, but they are definitely the ones, especially when you're getting started, you're going to be really thinking about. And there's a lot of variety in each of these categories to consider too. So when we think of grasses, I just want you to know they're fast germinating, fast growing, good at foraging nitrogen. And because they have these abundant and fibrous roots, those fibrous roots are actually good at opening channels in the soil for better infiltration. However, if it's a nitrogen poor environment, if your orchard doesn't have a lot of extra nitrogen, then they might not grow very well. They do produce a lot of carbon-rich biomass, which can add a lot of organic matter to the soil. But for the same reason, they're slower to decompose. That's a little bit of a concern more in nut orchards where you want to clean ground when you want to run harvest equipment. But in apples, you can really take advantage of the fact that they can build your soil with carbon, rich organic matter. And then there are some varieties, especially of triticale, that have a lot of root biomass, but don't produce a lot of above ground biomass. So if you have reasons you don't want a lot of above ground biomass in the winter, that can be really good for that strategy. Legumes, of course, the characteristic of legumes that makes them amazing cover crops is that they have these symbiosis with rhizobia bacteria, which basically grab nitrogen from the atmosphere and bring it into the soil through these nodules. And actually, it's an interesting thing. So you do need the rhizobia to be present. And so a question that comes up a lot is, should we inoculate rhizobia onto the legume cover crop seeds. In the trials that I ran for 3 years, we didn't inoculate, and we had really wonderful, spectacular nodulation. So my suggestion is, try them out without inoculating, unless the seed company inoculates them for free anyway. But you can try them without inoculation. See if you do not get good nodulation, then you can think about inoculating. So because they bring all this nitrogen in, they also are high nitrogen in general. The legumes are the biomass is high in nitrogen and therefore decomposes quickly. And that, once you incorporate that biomass, it also contributes nitrogen to the orchard. They're generally large seeded, which means that they can survive a little bit of drying out if the conditions are not perfect during the germination period. Clovers can be perennial or reseeding types, and they're low growing, so that's a nice option as opposed to some of the other ones like what we have here is basically field peas and faba beans or bell beans. Those are much more larger growing. And they do have flowers that are used by pollinators, so that's another advantage. The mustards, they are fast growing. They obviously produce lots of flowers that are good for pollinators. So they often predominate in pollinator focused blends. They have these large taproots that open up big channels in the soil. So you can see those large taproots there. And when you chop the cover crop and those roots die off, then those big channels get left in the soil. They can get very big if they're left, and if they're left very long, they can get pretty woody and slow to decompose. And also because they produce these high sulfur compounds, they can potentially suppress nematodes, especially if they're incorporated in a way that's called biofumigation. That's kind of a whole presentation in and of itself, but if nematodes are a concern, you might start researching more into that as an option. And then there are cover crop mixes, which is a way to hedge your bets. A little bit of everything. In case conditions aren't good for one thing, they'll probably be good for another. And that can be in-field variability or across season variability. And then you also combine the benefits that we just talked about from each of these types. And you also can balance carbon and nitrogen for good decomposition. And then there are pre-made mixes available from many seed companies. So you don't, although there are also some nice online tools if you want to design your own mix. Okay, so the cover cropping process is basically planting, letting it grow, and then termination, and those are seasonally. So planting happens, in the summer. The planting is in the fall, and then somewhere, by after December, between November and December, the cover crop's in the ground, and it's growing with winter rains, and then terminated in somewhere between February and May, which is a pretty wide time. And I want to point out, between March and April is when cover crop growth really picks up. And we'll talk about why that's important in a bit. I'm going to be going through this kind of quickly because I really realize my time is limited, and I do want to talk about addressing some of the concerns. But basically, planting, you need to figure out what you want to do and what you're trying to accomplish. Calculate the planting area, remembering that you're only planting the middles. You determine what equipment you have often determines how wide of a swath you're going to plant, although keeping in mind that we're trying to improve the soil for our trees, and so and we're not planting in the tree row. So the wider you can plant, the more benefits you'll see. But nonetheless, some of that will be determined by your equipment, and not only your, what planting equipment you have, but also what, like a mower you have, or how wide your disc is, and how many passes you're willing to make. And then select and order your seeds, and you can really, especially when getting started, really can work with seed companies. They have a lot of experience making recommendations. And of course, work with UC Cooperative Extension, Resource Conservation District. Often we have that expertise too. Okay, for planting, light ground preparation, a little bit of maybe a spring harrow, light disking. You don't need to till the ground as if you're planting corn or anything. So just a little, just so loosen the soil so you can have good seed to soil or soil to seed contact. Grain drills and no-till drills are great, but depending on your setup and your options, you can use a spreader. This is a really interesting setup with ATV with a little spreader on the front. But if you have a really small area you want to plant, these hand things that you just sling around your shoulders and hand spreaders can work too. I've seen that work remarkably well. Got to calibrate the equipment. I won't get into that too deeply right now, but yeah, got to make sure you calibrate it. And then after you calibrate it the first time, you really should be tracking how much seed you're using because the calibration is usually not exact. But you want to be able to use the seed that you're purchasing well and not have it all in one area and run out or have to run back and run over the area again. And then dragging or rolling you can see the seeds here in this photograph. They're just sitting on the soil. They're susceptible to birds or just germinating and not getting established. So a chain harrow is really nice for making that, covering them up, and a roller is another good option for just gently packing them in place and covering them so they're not exposed. Oh yeah, and then ideal to plant right ahead of rain, although we're planting somewhere between October and December, and I've heard in vineyards, for example, of people actually planting before harvest is finished, and the seed will kind of often sit there. If rains are late, you can give it a little bit of an irrigation if that's an option to get it started. But on the other hand, if you give it a little irrigation and they get started and then there's no rain, you can't have the seeds just die. So timing is really important. Ideally, someone once told me kind of poetically, it's like ideally you drive your last pass with your drill and the rain starts. Well, we actually had that happen this year, amazingly. So you can do your practices for keeping the tree row clear either before or after seeding. If you're spraying mechanical, I think that should work fine too, as long as you're not disturbing the middle. And then with walnuts, for example, that have large leaves, or we really want to get the plants germinated before leaf fall because leaves can smother them. I don't know if that's as much of a problem with apples, but something to keep in mind. It is ideal to get the seeds germinated and kind of a little bit grown before and we have typically what I've seen in California is leaf fall can be very late. So that shouldn't usually be a problem. And then you monitor winter growth, monitor for gophers and pests. I'll talk about this more, but if you've got gopher or ground squirrel issues, that can be a problem. And boy, am I out of time, Ellie, almost?
It's almost 3:15, but my presentation is actually a little bit on the shorter side. So if you want to take another
Okay.
5 minutes, that's totally fine.
Cool. I think we can get through it. So let's go for it. Thank you. So yeah, so you can also do your orchard sanitation and pruning, and then enjoy watching it grow. I think most people I've worked with who planted cover crops just really like to see it grow. I certainly do. So that's something you can do while it's growing. Now, termination. Time is of the essence. How come? So we want to accomplish maximum biomass to get those, biological and physical benefits of organic matter. But we also want to conserve moisture, especially in California, So timing termination where the cover crop has grown to a good size, but you're not drying the ground out is key. We'll talk about, you need to be monitoring soil moisture basically, and in multiple spots. Have some method, whether it's like, digging with a shovel or poking in with a tube or having soil moisture sensors. If you're doing it for maintaining pollinator and beneficials habitat, of course, you want to keep it through bloom. if you ideally, or as close to bloom as possible. That kind of goes, we'll talk about frost in a second. And then you want to manage the residue. I think that's more of an issue in nut crops where we don't want to have residue on the ground. But if you have concerns about having a lot of residue on the ground, then you want to be aware of how tough that biomass has gotten and how big. So termination is usually by mowing. You can also disc to incorporate it. Incorporation will help get that biomass more integrated into the soil. I think we found that if you just mow it on top, a lot of that biomass just decomposes and goes back into the atmosphere. But you do get the benefits from the roots still having grown there. On the other hand, it's just more work, and that mulch layer can be really nice for suppressing weeds as well. I won't talk about the roller crimper. That's an advanced method, but maybe if we do this again, we'll try to get into things like that. How about termination by grazing? As regenerative agriculture has become more popular, this is one of the big ones for orchards. It is a bit of an advanced technique, and I suggest you do a lot of research and talk with others, with folks who have practiced it or researched it. But it's definitely an option you can think about. And of course, that's before the crop. Sheep are generally used, and they don't tend to bother the trees, unlike goats, which would never be used. But nonetheless, generally this is done before the trees have leafed out or bloomed. And yeah, here you can see the difference. The front was grazed, the back has not been grazed yet. So that's another method of termination. Key considerations: soil type, water availability. We've touched on all these. Winter activities, pollinator support. Some people worry whether the cover crop is competing with their blooms for bees' attention. Generally, most studies suggest that it's actually the other way around. The bees are healthier, happier, and pollinate better. But some people get concerned, so that's something to think about. Frost risk, I'll talk about in a second. Pests like nematodes. Some cover crops can be a host for nematodes or other pests, so good to look into the species you're choosing and whether that might happen. Gophers and rodents, better to manage them before you start cover cropping if you have a problem. In the trials that I've had, I haven't seen them get up terribly bad. So something just to monitor when it's going on. And then you have to think about cost versus the benefits and maybe incentives that you have. I talked about, timely termination, monitoring moisture to modulate moisture usage. The other thing is I've noticed that when we have a lot of rain, the cover crops grow well. When we don't have a lot of winter rains, the cover crops don't grow so much, so they're less of a concern. You can just get through and mow them. Okay, frost risk is something I want to touch on because it's often mentioned as a deterrent. People worry that, there was a study that showed that bare, wet soil will absorb more heat during the day and radiate it back at night. And therefore, an orchard with bare ground will be warmer at night than one without. And this has deterred a lot of folks from cover cropping. This is a bit more complicated. You can read about it a little bit more, especially at the SAREP resources site. But I think we need to research this more. In fact, if you are in an area which is prone to frost and you have this concern, maybe we can do some research. So talk with Ellie, maybe, and we can reach out to me and see if we could. I'm really curious about this question. It seems to be a deterrent that's not very well kind of fleshed out, but it is a concern. And so that the suggestion is, if you're in an area that has this concern, and here's one, this is a vineyard, you just mow down to the ground ahead of bloom. So in Brentwood, where I work a lot, this is a cherry orchard. Frost is not generally a problem, and so cover crops are often growing while there's bloom. Okay, I'm probably out of time, so let me just say, what time is it? Oh, I'm way over time. So I am going to go, and I think I covered most of what I said. Tried out these cover crops. We're talking just for seed, somewhere between $13 and $35 an acre. You got to do the calculations for, fuel and labor. Okay, sorry, Ellie, I'm going to finish right after this. A lot of times question comes up, what about resident cover? And I say, okay, well, if you've got a nice resident cover, like this is Fillory or Cranesbill, that's pretty good growth. If you got that kind of biomass, although I would say you could keep your um, spray area strip a little narrower because that's not doing you a lot of favor where the trees are growing. But that's not bad. But if your orchards look like this, that's not a lot of biomass, not doing you a lot of good. Okay. Okay. And there we go. I sent you all the resources. I have lots of resources on video and at my YouTube channel. Do your research, start small, experiment, compare notes with others, and have fun. And sorry I took too much time.
Thank you so much, and no worries. My presentation is a little bit on the shorter side, so that's totally okay. Thank you for an excellent presentation. I really liked all the photos. It really helps to see what you're talking about with all the different types of cover crops.
Yeah, thanks
Okay.
for the opportunity. And I'll answer questions in the Q&A so if you have any. So yeah.
Thanks. Yeah, I see there is one question in the Q&A, and there may or may not be some in the chat. But yeah, if you could
Okay.
stick around and answer those, that would be awesome. Thank you
Super.
so much.
Thank you.
Okay, I'm going to go ahead and share my screen now. So I'm the next presenter. I'm also the host, as you know, but I'm going to be talking about apple orchard nutrients, irrigation, and dry farming. So as you know, my name is Ellie Andrews, and I'm the UC Cooperative Extension Specialty Crops Advisor for Sonoma, Marin, and Napa counties. So I'm going to start with a big picture overview focused on nutrient management. So we really need strategic, thoughtful nutrient management in order to maintain enough nutrients for healthy plant functioning in your orchard and good yield. So we want to start by getting nutrient status of your orchard into a healthy range. And then at that point, replace the amount of nutrients that you move out of the orchard at harvest in the fruit, because every time we harvest, we're exporting nutrients in that fruit out of the orchard. So it's important to replace those nutrients. So which nutrients do apple trees need? The answer is the same as all other plants. So they need macronutrients in large amounts. So that would be things like nitrogen, potassium, calcium. And then they need micronutrients in much smaller amounts. So that would be nutrients like zinc or boron. So typically, tree roots take these nutrients up and then move them throughout the tree through translocation. So another really important concept in nutrient management I think is super helpful is the law of the minimum. And this is just the idea that if one of these essential elements that I mentioned is low, plant health and yield will be low until that deficiency is lifted. And a lot of times this is represented as a barrel metaphor where you have a barrel full of water and you have a bunch of different staves. And here water is representing yield and the different staves on the barrel are representing different nutrients. And so here, in this image, you can see potassium is limiting the yield potential here. So supplying potassium in this case would increase the yield potential to the next most limiting factor, which in this case is nitrogen, just as an example. So I think that concept is helpful visually. And then another one that's nice is this curve that I'm going to show you on a plot where we have plant growth or yield on the vertical axis and then concentration of mineral nutrient in plant tissue, which is basically just the nutrient status of your tree on the horizontal axis. So starting way over on the left, we're increasing our, let's say, nitrogen, for instance, but we're still in this deficient range. So if you have nitrogen deficiency or any kind of deficiency, it's going to impair plant functioning and yield. And you'll see some visible deficiency symptoms that I'll go over in a moment. But on the other side, far other side of the spectrum here, over-fertilizing can lead to excess vigor, lower quality and quantity of yield, environmental damage, and ultimately wasted money. So we don't really want to be that far on that end of the spectrum either. We really want to be in this nice, safe zone where we have not too much and not too little. And you can use visual assessments and diagnostic tools that I'll go over to help you keep tree nutrient status in that safe range. So when we're thinking about rate and amounts, the amount of nutrients needed depends on a lot of factors. So it can be influenced by your scion and your rootstock, your tree age, your canopy size, just how big is your tree, how much nutrients does your tree need, the soil and plant nutrient status. So what's that existing status of your orchard, whether you're irrigating or dry farming, whether you have a high input system or a low input system, yield demand, whether you have a lot of yield that you're taking off, moving out of the orchard each year, or maybe it's a bit lower, and then the nutrient concentrations of the sources that you've chosen to use. So because that's a lot to think about, a really nice place to start is just with visual deficiency symptoms, which is personally one of my favorite topics. I really like this because this skill set allows you to go out and just look for yourself and see if you notice any of these symptoms. So I'm going to go over some important ones. There are more than this, but I'm just going to touch on the ones that I think are the most important for apple. So the first one is nitrogen. So this one's pretty straightforward. Nitrogen deficiency symptoms tend to appear on older leaves first as this uniform light green or yellowish leaves. This is also called chlorosis. And the key thing here with nitrogen deficiency is that the whole leaf is pretty uniform. You don't have splotches or patches, just kind of a pale uniform yellowish color. You might see reduced leaf size, thin, spindly, stunted shoot growth, short terminal shoots. And if it's really pretty severe, older leaves may also die. You might see reduced bud formation and fruit set, small fruit size, and fruit that mature early, and ultimately reduced yield. On the other hand, like I mentioned with that curve earlier, we also don't want too much nitrogen because that can also cause problems. So too much nitrogen can cause large dark green leaves that remain on the tree in the late autumn. It can cause just too much foliage, vegetative growth, increased susceptibility to fire blight because fire blight travels along with that increased shoot growth, unfortunately. It can cause reduced fruit quality, poor color, fruit that lose firmness faster in storage, and things like that. So again, we want to be in this nice sort of middle ground where we have just enough nitrogen, but not too much. Moving on to potassium. So potassium deficiency symptoms look more like this, where they appear on older leaves first, but can affect young leaves too when it's severe. You're going to see yellowing of leaf margins that can turn brown over time and curl upwards. This might appear more as fruit reaches maturity because the fruits actually accumulate a lot of potassium themselves, because potassium plays a lot of important roles in fruit development and cell expansion. So you might kind of see this around fruit or as fruit size up. And potassium deficiency is more common in sandy soil than clay soil because sandy soil has lower cation exchange capacity, which basically just means that it can't retain or hold on to cations like potassium quite so well. So more common in sandy soils. So another really interesting one is calcium deficiency. So this shows up as something called bitter pit, and at first glance, you might think it kind of looks like a disease, but this is actually calcium deficiency that causes lesions, often concentrated towards the calyx end of the fruit. And it causes these small, quirky little brown areas on the fruit, making it unmarketable, of course. And symptoms might show up a few weeks before harvest or in storage. And this problem is often also related to low water uptake as well, which is just good to bear in mind. And the last one here, I'm going to mention zinc. This is probably a little less common in our area, but it can happen in apples and in fruit trees. So zinc deficiency symptoms look like small chlorotic or long leaves, sometimes interveinal chlorosis, and this rosetting that's caused by short internodes. And you might see some blind wood on newer shoots where there's no leaves. So all of those visual signs are really helpful, but how do you know for sure if your trees have nutrient deficiencies? Really, the only way to know 100% for sure is if you collect some samples and send them to a lab for leaf tissue testing, which is a really, really helpful tool. So leaf tissue analysis tells us the concentration of nutrients in apple leaves. And this is the most reliable direct assessment of tree nutrient status. You do this, just once in July is great, and then you send it off to a lab that offers the service. And if you want to get into this in more detail, there's a really nice write-up on the CDFA website, and I'll send a link to this out in the follow-up email as well. But this goes into it in more detail. So after you do that, you'll get your results back, and then you can compare the numbers that you get to the recommended ranges specifically for apple trees. And I really like this because it's a pretty simple quantitative assessment that anyone can do. It gives you numbers, which is nice. And, this allows you to answer that question, are your results within the optimum ranges, that safe zone? And if not, then you can use this information to guide your management decisions with fertilizers, amendments, and stuff like that. And while you're at it, I think it would be great to do some soil and water sampling if you've never done this before. This is really, really helpful to help diagnose issues prior to planting, but also over time. I know some folks do this every year in the fall, others maybe every couple years, just depends on what your questions are. But this helps you understand nutrient availability and soil constraints at your site. It provides complementary information with leaf sampling and just gives you a fuller picture of what's going on with nutrients. And it can also help guide fertilizer and amendment applications. So your big picture goal here is moderate tree vigor with minimal but sufficient nutrition. So enough but not too much. And you want to match timing of nutrient availability with crop demand, making sure those nutrients are in the root zone where trees can take them up. And just a little more information on some key nutrients here. For nitrogen, you might not really need to apply it every year. This is what I'm hearing from apple growers in our region. But you can apply it as needed, really, based on vigor. So your goal is around 18 to 30 inches of shoot growth on young apple trees. You want to see healthy bloom and fruit set. And of course, if you really want to, you can use leaf tissue results as well to help guide that. And so it might look something like this, where you have cover crops in the spring that you disc in. Maybe you might apply some organic fertilizers if needed throughout the season. And a lot of folks in our region use compost applied in late autumn. And I know some growers here really just rely on compost and cover crops, and sometimes that's even more than enough. So it depends a lot on your situation and what rates you choose and that kind of thing. But this is something that you can fine tune over time. And with potassium, I think it's important to bear in mind that those leaf potassium deficiency symptoms can look a lot like severe water stress symptoms too. So, and it's often these two things are happening at the same time. So this could be related to low water uptake. If you know, because you did those soil samples, that you have plenty of potassium in your soil, you could try increasing irrigation first just to see if that helps with tree uptake. And bear in mind, if you have sandy soil, high rainfall, like, the last couple years we've had a lot more rain out here, which has been great. But if you have sandy soil and you also know that you have a potassium deficiency, you might want to think about applying potassium in the early spring rather than the fall, just to avoid leaching over the wintertime. For calcium, relying on visual symptoms, which are pretty characteristic, and then also leaf tissue samples, if you want to go that route, it can be really helpful. Looking at your soil test results, if you see low calcium and low pH, you could apply lime to help with both of those things. But if pH isn't really so much of a problem, you could just use a calcium sulfate fertilizer. And then again with zinc, these symptoms, you can look for the rosetting and the little leaves. And if you need to, if it's really important, and especially if you did those leaf tissue tests and have seen, okay, this is definitely a zinc deficiency, you could consider foliar applying zinc because it's not particularly mobile in the soil. So this would be done in the late dormant period. But I just want to emphasize it's very important to follow the labeled directions really carefully because it can be easy to over apply foliar nutrients and burn your trees just from toxicity or too much. So whatever you decide to use, just make sure you're reading the label for that specific product. So moving on to water now, I'm going to go over a couple quick strategies here that will support you in climate resilient water management. So the first thing is, if possible, it would be great if you have access to several sources of irrigation water. You want to do regular irrigation system maintenance. You want to make sure you're matching irrigation duration and frequency with tree needs. You can think about phenology-based deficit irrigation. A lot of farmers in our region use dry farming, which I'll get into in a little more detail soon, and soil health practices can really help improve soil, water, plant dynamics. So for some brief context, I know a lot of folks here are from other areas besides Sonoma County where I'm based, but here we have a lot of different rootstocks and scions in the same orchard, which is amazing for a lot of reasons, that diversity, but it can create some irrigation challenges because these different rootstocks and scions, there might be different water responses, different needs, different degrees of drought tolerance. And I would say probably around 95% of apple orchards here are dry farmed. So that means they do not have irrigation set up at all, while we do have a small fraction that irrigate. So that's just to say there's a lot of variety. So I think it's important to choose the water management strategies that make sense for your situation, because there's really no one size fits all. Okay, so regular irrigation, if you're able to do this, if you have water access and your system set up, this supports good growth conditions and healthy fruit trees. Supplying water helps prevent severe water stress, which can permanently damage trees. So you want to start also by choosing cultivars that have relatively low water requirements that you know that you can meet as much as possible. So here are some visual symptoms of tree water stress. You're going to see loss of turgor pressure, which is just leaf wilting, marginal necrosis, that dead leaf tissue on the margins or on the edges, bark cracking, stunted growth, defoliation, and if it's really severe, it can cause tree death. Water stress can also lead to fruit sunburn or increase susceptibility because of higher fruit temperatures. And it can increase susceptibility to sunburn on branches and trunks too. So if you're able to whitewash your trunks and large scaffold branches, that can really help. So rapid cell division happens in early fruit development. So receiving enough water at that point in time is really important for fruit size and quality. But it's important to bear in mind that your goals for fruit size and quality and flavor may really differ depending on your end use. So if you're growing for fresh market versus cider apples, for example. So now we're going to get a little more into how much water you need. So to start with, as I'm sure you all are familiar with, this term evapotranspiration, this is just daily water use. So this is the amount of water that's going into the atmosphere that's transpired by the plant and that evaporates from the soil surface. And this amount of water that the trees need and the system needs depends on a lot of things. So it depends on your tree size, tree age, rootstock and scion, tree spacing, soil type, your location. You know, we have a lot of microclimates in our region and other environmental factors. So you probably noticed some similarities between this list and the list for the amount of nutrients that trees need as well. So there's a lot of things to consider. But the most direct way to measure tree water stress and really answer the question, how thirsty are my trees right now, is stem water potential. So stem water potential is a way to collect data at midday or early afternoon, and then you compare that information to established ranges for apples. So the first thing you do is you have this little bag that goes around a leaf, and that pauses transpiration. And so you wait 10 minutes. And then you pick the leaf and you can put it in a pressure chamber. There's a couple different types out there. I know orchard managers find the one on the far left a little bit more convenient. It's just basically the same kind of model as a bike pump, but there's a little chamber and a dial on top. So you place the leaf in that chamber. There's also a little fancier, higher tech options like this one here where we have a nitrogen gas tank. But in either case, you're putting this leaf in a pressurized chamber, pressurizing it very slowly and looking at the petiole. And once little bubbles come out, then you say, okay, yeah, and you stop pressurizing it or increasing the pressure, and you look and see what number you have. So if you get somewhere between -6 to -10, your trees are going to be more or less close to fully irrigated. If it's anywhere from 10 to 20, then that's going to be slightly to moderately water stressed. And if you have values that are lower than -20 bars, then you're looking at some pretty severe water stress. So another option out there, I think this is probably maybe more relevant for larger scale growers, but microtensiometers and dendrometers are sensors that are embedded into trunks, and they continuously measure tree water tension. So more tension means more stress. And this is nice because it's less time intensive than a pressure chamber. You don't have to do nearly as much, but it is a bit more expensive. There are all sorts of soil moisture monitoring tools out there as well. Here are just a couple examples. And these will tell you the current status of the amount of water in your soil. There's also this great approach that I wanted to mention to folks called phenology-based deficit irrigation. And this is just the idea that we want to match the timing of water deficit with the timing of crop water stress tolerance. So here we're just applying water during periods of critical plant growth and development, but not during water stress sensitive stages. So we're using phenology to kind of guide that. And this allows us to reduce water use during periods of plant growth when crops have lower sensitivity. So what does that look like in apples? Bearing trees can tolerate some moderate water stress until the fruit is about half size, and then irrigation can help increase fruit size. But then moderate water stress during later fruit development can increase fruit quality. So regulated deficit irrigation can reduce fruit size. That's something to bear in mind for folks where fruit size matters. But it also can increase fruit polyphenolic compounds, total soluble solids, firmness, titratable acidity, and broadly speaking, it's probably going to have neutral or positive impacts for cider quality. So wrapping up, I'm just going to spend a couple slides on dry farming because this is a really common practice in our region. So this is unirrigated crop production during the dry season. So you usually see this in regions receiving at least 20 inches of rainfall each year with the warm summer Mediterranean climate. So in dry farming, soil moisture from the rainy season is captured and stored in the soil and then used by crops later during the dry season. And there are various strategies that you can use to help make this effective. So some quick benefits of dry farming. The biggest one, of course, is water savings. So this allows you to reduce irrigation use and costs. This can allow you to increase productivity on land with low or no water access. It can help you mitigate drought-related risks, offer some crop security during times of uncertain water supply. A lot of folks report reduced weed pressure as a big benefit of dry farming, improved yield quality, and potentially new marketing potential due to quality improvements and water conservation. However, there are also some limitations, of course. So yield quantity tends to be lower than irrigated orchards, so I think it's important to set realistic expectations around that. You might want to space the plants out a bit wider so that they're not competing for water. And some winters, we know that it might not rain at all here, which can lead to more severe water stress the next year. So how do growers do this or optimize this? A lot of folks in apple orchards here use dust mulching. So this is just a practice where you're using some surface tillage to seal in moisture by breaking capillary action. The benefit here is moisture retention deeper in the soil, but of course, the more you disrupt the soil through tillage, the more you increase erosion risk and over time reduce soil organic matter. So this might be more suitable in some locations than others. And there are other strategies you can use like rootstock and scion selection. You want to think about water efficient, drought tolerant, early maturing options with deep roots. You want to improve soil structure for good infiltration. And yeah, that wider tree spacing, like I mentioned, can help. So just bear in mind, dry farming, it's not all or nothing. You can consider trying this out first on a small area, hone your approach before scaling up. Some areas of your orchard might be better for this than others. And you can deep irrigate your crops in at planting to really help them get established. And you could even irrigate at key times throughout the dry season if needed, especially when you're just starting to experiment with dry farming. If you're really curious about this, Oregon State University, they have a dry farming institute, and they actually have an annual virtual convening on dry farming in February. So coming up next month. And there's also an online accelerator program that is free and self-paced that anyone can take. And I just wanted to mention, a couple UC Apple resources that have a lot of information in them, including things on irrigation and nutrient management, soil management, the organic apple production manual, and the cost and return studies are really insightful as well. So thank you all for your time. And I know it is 3:46, so I'm actually going to answer questions in the chat and in the Q&A separately, so we can go on to the next presenter. Okay, so I'm going to make our last two presenters co-hosts. Welcome.
Hello, everybody. I'm Stefano.
Hello, welcome. Thank you so much for being here and joining us.
Thank you for the invitation.
Yes. Oh, absolutely. Yeah, very grateful for your expertise here. Are you able to share your screen? Do you want to go ahead
Sure,
and give that
I
a
will
shot?
start immediately. Yeah.
Okay, thanks.
Can you see the slide now?
Yes, that looks perfect.
Okay, so I will start my presentation. So looking at your orchard, the picture that you just show us, I think that you will see something a little bit different from my presentation, but I hope that will be useful in any case as a generic information. Okay, so I always start my presentation on training system and orchard management, considering that we have to manage many things when we have to decide what to do to establish an orchard. And for example, we have for sure the genetic component that is cultivar rootstock and the interaction between the cultivar and the rootstock. And on top of that, of course, we have we have to know all the environmental conditions. So the soil, like you pointed out very well, is very important because some soil retain water in a different way. The level of weather that you can have it, especially rain, of course. And then how you manage your soil. So, because if you have water is one story. If you don't have it, it's a different story, unfortunately. So, and then you need to know how to prune, how to irrigate and everything. So when you know all those information, normally you decide the training system and the planting distance. So that is the scheme that normally I put in place every time I have to decide to establish an orchard. So I will go through some of those factors, cannot cover everything, but I will start with the cultivar because not all the cultivar are the same. They have a different habit. This is an old work done by a French person called Lespinasse, that in our area is a kind of a legend. He defined four different type of tree: type 1, type 2, type 3, type 4. Long story short, the type 3 is like a golden, very easy tree to train, very easy tree to manage because it's producing mainly on all kind of wood. When you go in a type 4, that is more a granny smith, you have a lot of blind wood, the cropping area is moving out, become a little bit more complicated. And I will tell you something about how to prune those kind of trees. So, but the habit of the variety can be completely different. This is a nice slide that show you how the habit of the variety can change from like a red chief that is a spur type tree to the most acrotonic one that is number 20 in the bottom corner of the slide. So, but all those habits are present in the varieties. And then the other thing that you need to know, of course, when you define a training system, you need to know the peel, the color, because if you have like a bicolor variety like Gala, or that need light and the fruit need to be exposed to the light, you need to expose the fruit. If you have another variety like Granny, for example, that is green, of course, you need to more to shade the fruit because when the yellowish color come up, so normally they don't have a great value for the market. If you have different situation like a yellow apple or a red apple, is a little bit easier than the two conditions that I mentioned before. So why expose the fruit to the light is important because sometimes you need the light to develop the overcolor. So, and you can see here just the effect of the leaves on the fruit. And unfortunately, Washington, I come from Washington, everybody know that we have a high standard for quality. If you have fruit like that, they will never meet the fancy quality category. So, and then the variety are so many. You have to imagine that actually we grow more than 30 apple variety. They are all different for the habit of the tree, for the color of the skin, and match them with the rootstock sometimes is complicated. So if you look here, we have I modified this scale of vigor from the Cornell, rootstock scale of Gennaro, and we have a lot of rootstock, and probably some of them will be very useful for your dry condition in California. We have a lot of them already here tested, and in any case, the combination between rootstock and variety determine everything about your training system. So, but there is something that we know quite well, that a dwarfing rootstock is important and necessary, at least for the high density planting, because when you look at the amount of dry matter that the tree is able to produce, a large portion of this dry matter is diverted to the fruit. And you have, for example, if you look here at this chart here, the dark blue, 70% of the dry matter produced on the tree go to the fruit, 9 to the leaf, and 16 to the wood. If you have a much vigorous rootstock like MM106, only 45% of the total dry matter that the tree produces is for the fruit. And then you see that the amount of dry matter used to grow the structure of the tree become high, and you have a lot of vigor in many cases. So I think that we are facing in many cases excess of vigor. That probably is the opposite problem that you have in a dry cultivation. And of course, with the progression of the age of the tree, of course, the diversion of dry matter to the fruit become quite important. As you can see here in the bottom, you have the trunk circumference and how the tree grow by time. And here the percentage of dry matter, and you see how the dry matter increase to the fruit. Okay, so when the tree complete the growth. So, but this is also the trick of crop load, especially if you are in a dry condition. If you crop your tree too early, then the tree will stop to grow because there is no more dry matter that go to the structure of your tree, but go mainly on the fruit. So looking at that, I put together some training system evolution. I don't want to cover all of them because it will be probably not a 20-minute presentation, but much longer. But we have a 3 category of training system. So the one in the volume, like vase, pyramid, central leader, very popular in old orchard here in Washington. And then we have the most innovative, let's say, spindle type that are conic tree. And then we have a new tendency, especially for the mechanization of the operation and with the automatization probably that we can have in the future, hopefully. We go more on a fruit wall type and a biax tree. And I'm going to explain a little bit what is the thing that we are trying to do here. So, okay. Another thing that we need to understand is the tree architecture and light interception. I prefer mainly to the 3 type of training system that are quite popular in Washington. That is on the left is a spindle, Here you have a fruit wall, and then you have a V system there. And the light interception is quite important because more light you intercept, more efficient normally is your tree, and better is the quality of your fruit. And how you can increase light interception, you have to increase tree density per acre. Otherwise, you have a lot of area that the light hit the ground and is lost. And of course, you have to optimize the leaf area index. That is a very important index because it's the area of all the leaves divided the value of the area that is assigned to the single tree. So this value can be low or high, but if it's too low, you are in trouble. If it's too high, you are in trouble for the opposite reason, and I'm going to show you why. So up to 50% normally the light interception and the yield is linear, as you can see here, then become a little bit erratic because there are other components that come in place, especially because light interception can be not optimized because in the canopy you have large area of those canopy that can be in the shadow. And this is why it's important that we know that something can happen if you are not in the optimal range of 65, 75%, because when you drop to 50, 55, you affect the quality and the growth of the fruit. You go below 35, you reduce the export of the carbohydrate from shoot to fruit. And below 20, you can thin. So this is another thing that is important in the large canopy when all the inside area is covered by leaves and there is a lot of shade. Normally, you don't have fruit production there. So how you can maximize the yield and fruit quality? Of course, you need to have a well open exposed canopy, avoid the canopy close the coverage too early, and have a good exposure to light. And if it's not happening, you can summer prune. In your condition, probably you need to be very wise because also in our condition, summer prune done too late or close to heat wave or when there is too much hot condition, you can sunburn almost all the fruit if you don't have any mechanism to protect them. So, but this is the value that normally we consider optimal for the leaf area index is from 1.1 to 2, and some you can go up to 2.3. But if you go too high, mean that your canopy is in the shade. If you go too low, mean that you don't cover enough and your tree are not don't have enough leaves. So those are the parameters that normally as a physiology that work on training system, we take a look. And probably now I'm going through some of the system that we have. I describe a little bit three systems, mainly one system from France that probably can be suitable for your condition, and spindle and biax. So the one that I mentioned is a Solax, and is a very good system for type 3 and type 4 variety, and for low density planting. Okay, so probably not low like the picture I saw from Ali, but it is still good. And it is a way that you can have to put the fruit exposed to the light, especially if you have a bicolor variety and you need to develop an overcolor. Otherwise, the market doesn't pay any money for not colored fruit. You have to deal with large branches normally, and seeing them manually. Sometimes this can be expensive, but is a system that can help, especially because look at the fruit that we collect, for example, in the top, in the middle portion, or in the bottom. So these are the kind of fruit, the one in the bottom, that have no market almost. At least the quality is really low. So we need to minimize this kind of situation. And if you look here, this is you don't have to look at the chart in general, but those are 3 years in the road. And the red is Pink Lady in Solax system, and the dark green is Pink Lady in the spindle. So the other one is crisp pink, that mean that they are not colored fruit. And what you see that in the Solax, normally the amount of pink lady and the yield increase because of how the tree is strained and how the fruit are exposed, compare the spindle that show to be quite stable in this case and lower compared the amount of fruit that you can have on a Solax. So it is why I mentioned it's not very popular in Washington at all, but in Europe, they're using quite a lot. And then we have the typical spindle tree for apple that is probably the most important with the V trellis system we have in Washington. And this is an example of Fuji, second leaf. You can have a very well feathered tree from the nursery. And in this case, you have a very easy crop in the second here, but everything need to be almost perfect to do that. Okay, you need to have you can see here irrigation, quite fertile soil. So this is a optimum level of management and technology. So everything need to be perfect to have such kind of yield in the second year. And of course, something about the pruning, about the spindle. Normally, the branch need to be tied down, and the tree looks like that in the progression of the year from the planting to the fourth year. But the problem is that now, with the labor cost and everything, I think that nobody can afford really to tie down any single limbs in a tree, at least in Washington. And especially for variety that are type 4, if you tie down the branch, you just make the situation worse, because if you can see here, you just develop blind wood in this case. And this can be really not great, because you are moving out of the tree and of the space that you assign to the tree, the cropping area. So, and I want to point it out something that probably more and more variety have this kind of characteristic. This is a 1 year old shoot of the previous year that have already flower bud on. And I call, with an Italian word, I call ramo misto. And because I didn't find a definition in English, but what I pointed out that all the bud in this case of the tree are like a population. So you need to think about that not all of them bloom at the same time, because they are not being formed at the same time in the previous year. So, and you can see here, for example, that there is at least a 7 days difference in the blooming of the ramo misto that you can see here. And if you look a little bit on the main axis here, you saw that the flower developed from a spore. They are already end of bloom. So, and this can really be very problematic for the thinning or for the fruit set and everything. So there are so many things that we need to take in account about the habit of a variety. And it is, for example, at VAS 38, the most important variety, probably you know, as Cosmic Crisp in Washington. And this is an example of how the ramo misto evolve. And you see that there are a lot of blind wood in any case, formed, completely not formed spore here. And then only the one that the really end that they set and give you a shoot, that is a normal situation for an apple. And in many cases, you have this situation here, that the size of this apple is much bigger than the one that developed on the lateral bud of the ramo misto. So this is a huge impact on fruit quality. Okay, so that is quite important. And here, like in the letter C, you can see here that this spore is completely extincted. There is nothing that can come out from here. No shoot, no fruit, nothing. So, and this is the evolution. So you look at the shoot like that, the following here, they look more or less like the picture above. And how to manage things. We start, at least in my program, we suggest the click pruning. That is something that has been developed originally for me in 2 different crop, pear and cherries. And it's quite working well to minimize the blind wood and help to build ramification in the branch. And the other thing that I want to cover is also in the spindle, you have some need of maintaining a thin canopy, because if you look at this graphic here, the dark area is the one that received less than 30% total light. And normally with this amount of light, you have no bud formation. So normally, and this is quite important because you can end up also in a situation like that is not just that you don't develop flower bud, but the fruit look completely different. Look, this one is growing in the shade and is completely green. The one on the top is completely ready, already sunburned in our condition. So you end up having a gradient of maturity in your tree. So what we can do to improve that? Normally we use extend day mylar, but it's quite expensive, and in any case, the extend day can be used more than 1 year. Mylar is a single year use. So for this reason, from long time ago, I started to work on fruit wall and bi-axis and canopies, and the fruit wall starts long time ago. In Italy, we have a lot of palmetta, and palmetta is a flat canopy, but really tall. And if you see here, this is a nice work done by Terence Robinson and Alan Lasco that prove how the light distribution is. And you see that in the bottom, the percentage of the light is quite low compared to the top. And again, so this is the point when your tree gets sick and too tall, then you develop a situation like that where the bottom is less productive, the middle is okay, but you got large part of the fruit on the top. And this is how the energy distribution is. So it's more on the top and in the side than in the bottom, because the large part of the day, the top is shading the bottom of the other row. And for this reason, we develop in Europe a different approach that is mainly related to the fruit wall. And in this case, we are not speaking anymore for fruit for tree, but fruit for square meter. And this is the amount of fruit that you have per square meter, that is 25. And theoretically, this is the amount of fruit that you can have in an orchard that is very well trained and grow in optimal condition. So, and this is also how you minimize, for example, the problem of the shade that we were mentioning. This is an orchard in Washington, and you can see that the canopy is really thin and the trees are planted at high density. So the point is don't shade your trees, otherwise the crop will be not of the top quality. And how we can manage also by axe tree, for example. There are three main options that you can have it. And the first one is you can grow the tree in the nursery. You can head back a tree after planting, or you can top graft and change the style of an existing orchard. So, and I'm going to cover the different situation. This is how you produce the tree in the nursery. You can do a double chip budding, like the first case here in the top, or you can just head back the tree and grow two leader, but one become always a little bit bigger than the other. I don't find any difference, honestly, in the size, especially if it's an apple, because normally large apple are mainly on less vigorous branches. So, and I make always this joke with myself and all the one that think that we sometime we reinvent the wheel. This is a publication of Robinson, not our friend in Cornell, but a previous one in 1878, and this comes from the library of Cornell, where you can see here a double axe done in France a long time ago. So we just change the technique and then we go through the things. So, but anyway, so this is how normally I suggest the grower to build a bi-axe in the orchard. You head back, I head back 2 times. The first a little bit higher when they are the shoot that want to replace the leader. I do a second cut and I select 2 shoot from the bottom with a much better angle. So this give me a very nice angle at the bottom and a great verticality of my axe. And this is more or less a scheme how to manage the things, remove the competitor tie. You have to have 1 Y here, and then you grow small branches, normally 20, 25 branches per each of these axes. And this will give you a very easy tree to manage in a fruit wall condition. So this is an example, possible mistake. This is not good. Here, someone used a branch instead of using a bud from the stem, this is the right condition for biax tree, okay? And in case you want to do a top graft, top working, you can replace an existing orchard. And this is a trial we did in Washington where we replaced with a single, double, and triple axe. That is a single, this is a double, and you can see here, this is a triple. So you cover the space very quickly here, and you enter in production very quickly with this system because you have a very powerful root system, and you fill up the space very quickly. That is the problem number one we have in Washington. That is the bloom. And ideally, as I say, you need to have 20, 25 small branches per each axis. And what is nice, and if you look at this graphic, the different zone of the tree, you can see that the red bar is the spindle, and here you have the number of shoot, and you can see that almost you double the number of shoot in a biax tree compared to spindle. But when you look at the length of the biax shoot is almost half. So if you want to reduce the vigor, this is a very good situation. So, but if you have already a vigor, a low vigor like a bud 9 and a dwarfing variety, don't even try to do a biax because you just make a mess because everything will be not growing and everything will be a flower bud. So the effect of the biax on quality is clear because you expose the fruit to the light. And I want to just point it out here that also the distribution of the fruit is more homogeneous. If you look at this graphic here, you can see that the biax is almost 50-50 between the bottom and the top. When you look at the spindle, you have only 26% of the fruit in the top and 73% in the bottom. And the bottom normally is when you have some shade and the color is not great. So if you have a bicolor variety, keep in mind this kind of information. And again, also the biax can change a little bit where the fruit to born. And if you see here, if you compare the different wood that support the fruit, you see that the spore on axis, for example, are much more present on the biax, 70% compare the mix that I was showing you a little bit at the beginning when I was showing the ramo misto. So this is the condition you had. You have a much more homogeneous fruit size and bloom time. So pruning, you can do scoring. So this is something that is quite efficient for us and is quite important. The scoring is okay. Be careful with far blight, but normally we do in March, and this help a lot to minimize blind wood. Oops. And this is the result. And remove big branches that are over 30% of the size of the trunk where they're inserted. We do a lot of head cut or the click pruning like this one to minimize the type 4 variety blind wood, and it's working quite well, at least in our condition. And then I have a little bit on mechanical pruning. I will be a little bit longer. I apologize for Greg. I will try to be faster now. But mechanical pruning for the fruit wall is great operation. You need to be sure that your system doesn't fail because you want a vertical tree. Otherwise, you end up in a situation like this one that cannot be pruned. This is a typical orchard that can be mechanically pruned. And you use a lot of summer pruning in this case. We use a lot in Washington, summer pruning in June and July. And sometimes we prune close to harvest if the temperature allowed us to do that. There are different machines. So there's a saw circular disc or front looping machine with a bar. And this is quite efficient, and we use a lot this kind of machine. And this is another prototype from Italy that has been very powerful use in Italy, this kind of machine. And this is the kind of intervention that you can have. It cannot cut more than maximum is 3/4 of an inch size branch. Otherwise, you break a little bit the equipment. So it's very important to summer prune because you have a positive effect on flower bud induction. And normally you can convert vegetative meristem into generative meristem, like I was showing here. And what happened that when you stop the growth, you have a cut here. There is a kind of ethylene production, and if you are not if you are doing that in the right time, you develop a bud behind the cut, as you can see here. If you are doing something wrong, or there is an excess of water that probably you will not have, you end up having a regrowth, and this spoil completely the situation. This has been done late in August when the tree are entering in a second flush of growth, and this is a disaster because then you end up having all the leaves still hanging in winter and a lot of damage from frost. Okay, so be careful if you want to try something like that. And this is another example of how all the young shoot has been killed by the frost. And some picture of the wheat rallies, that is an iconic training system we have here in Washington. It's very nice until it's not. If you can you see the line I draw here and the fabric that they're using there, because is already shading. The shade is the enemy of any training system somehow, because if you want to have good fruit, they born generally where there is the light, and you can see here and here and here that the shade can be the problem. So if they get too close, then there is not enough light to promote the quality of the fruit. And I dedicate last minute of my presentation on planar canopy. Probably is something that we will see in the future. There is large movement of people thinking that this is the future of the training system, and especially in New Zealand, a very nice group of researchers. Stuart Casting was a pioneering guy that developed the things, and then had other researchers follow. And they are using this multi-leader system that honestly, it seems innovative, but for me come from the past. In the same book where I found the biax, I found also the multi-leader. So, and it is very funny because now we are just planting really narrow. It is 1.5 meter is 5 to 6 feet distance. And with this training system, of course, and the condition of New Zealand, you can, this is the same multi-system leader in Ferrara, and they're sprayed this kind of things everywhere. If you do well, it can be very productive, but at the same time, you need to manage everything really, really in a nice way, because otherwise, this is a perfect tree. But if you look, then you have other picture that the tree doesn't look so nice. And they were publishing that the yield in a case of when you compare with a traditional spindle is completely different. At the beginning is lower because they are training all those leaders, but then you see that by year 6 they overcome the spindle, so quite definitively. So, but this is where I'm not sure, because those 6 years for me are also difficult to manage. Another system that has been developed in Italy by a colleague called Dorigoni is Guyot, is similar to what you grow for grape. And they prepare the tree already with this reversed gradient of vigor in the nursery, in the way that when they plant, they have the shoot, the longer shoot in the place where normally the shoot grow less. And this help a lot to minimize the variability. But again, if everything goes well, you have a situation like that when you do a multi-axis. You see that there is always a gradient of vigor in the axis. You see already that there are some window here because or there is a lack of a shoot, or sometimes they have to remove it because it's too vigorous. Can you see? Now they're growing not an axis, but another tree on top of the one that is being bended. And all this vigor is taking out all the vigor of the other one. So need to be cautious about that. And then you can end up with something that is not even close to the previous picture that I mentioned to you, or something like that. You can see, and there is a lot of variability. So, and you will never fill the space, especially for the reason that I told you before, because when the trees start to crop, the dry matter go to the fruit and not go anymore to the fruit wall. So if you try a multi-leader, be sure that you fill up the space before to crop. Otherwise, you end up with a huge amount of variability. With that, I end my presentation. I'm 3 minutes longer. I apologize for that. If there is any question, I will be happy to answer, or probably you can write me. Thank you.
Thank you so much. That was so informative and very helpful for our region. I really appreciate it. It's great to see what all
But you are in a dry orchard condition.
Yeah,
So it's
yeah, it's true.
complicated.
And there is a question actually in the Q&A about that, about, if you were planting in a low water area, and so you can read that question there.
Yeah,
Just for the sake of time, I think we should probably move
yeah,
on to Dr. Pex. But
sure.
yeah, thank you so much. I really
Take
appreciate
care,
it.
and thank you for the invitation. Thank you.
Yes,
I
absolutely.
I have to leave, Kelly, in any case, so I cannot stay. So
Okay. All right. Well, welcome, Dr. Peck, our final presenter. Thank you so much for being here all the way from New York. Our other presenter from far, far away outside California. So I'll hand it over to you and take it away.
Awesome. Thanks, Ellie. Sounds okay.
Yes, sound is good.
And video is running.
Yeah, that looks great.
Great. All right. Well, hey, thanks for having me here today. Yeah, a little later for us in New York, but that's okay. I'll dinner afterwards. Um, but really happy to be here. I actually have connections back to Sonoma County. By late 90s, I worked at the Sonoma Antique Apple Nursery, now defunct, and they sold the business to Trees of Antiquity that are down in Santa Barbara now. But that was my kind of first foray into apple production, and just a fantastic place. And was living in Healdsburg and enjoyed it. Spent several years in the Bay Area, and met my wife down in Santa Cruz. So love California. Maybe one day I'll be able to go out and visit Ellie in person, and come and see some of the orchards that are in her region. So she asked me to come in today and talk a little bit about what we're doing with cider apple production in New York. And so really glad to be, here we go, part of a really large team of people at Cornell who are working on cider, largely because we have big apple industry and we have a big wine, grape, and wine production industry here. So cider pulls from both of those areas in order to gain that expertise and build a program. So I'm probably, one of the few people in the country really who get to dedicate as much time as I do to the study of cider apple orchards. But because of all these people at Cornell doing research, just really fantastic, and it's really aided what we can do. I'll mention also I teach an undergraduate class. It's going on right now with co-instructor Kathy Arnick. We have 120 undergraduate students taking our cider production lecture, which is fantastic. They get to taste ciders. We have special permission from the state of New York to serve alcohol to minors as long as it's for educational purposes. We do a lot of outreach too. I work with the Cider Institute of New York, Cider Institute of North America, and do presentations for them. And then also we have a program work team or a working group internally, and we lead summer tours for our producers in different regions of New York so that they can learn from each other, see what's going on both in the orchard and in the cidery. So we have a commercial lab, we have a lot of research orchards, we have a craft beverage analytical lab, and we also have access, and I'll mention it again a little bit later in my talk here, to the USDA's apple collection, which is in Geneva, New York. So it's about 45 minutes north of me. Okay, so after Stefano's presentation, really don't need to go into much detail on this. I think this was already covered in really excellent by Stefano, but I'll hit on many of the same points that he did, but just trying to apply those instead of for fresh market production, where it's all about getting large yields, getting that fruit color just right, and really trying to make the system efficient for fresh market. I'll be trying to apply some of those principles to the production of cider. And so we look at many of the same elements, right? Cultivar and rootstock selection, orchard design, how do we manage the crop load, how are we managing nitrogen? And for cider apples, one thing that's quite different is how are we harvesting the apples? I think a lot of cider apples, if you're not familiar with the industry, a lot of people have this idea of cider apple orchards looking like this image on the top, and certainly this is possible. There's a picture I took myself in Somerset, England, just a few years ago, and they're grazed orchards here. In this case, sheep. Sometimes it's cattle. The trees are budded really high. They use an inner stem here to allow for these tall trunks, and then the tree canopies above. Not a very efficient system in terms of productivity, like we would see in these bottom orchards. This is one of my experimental orchards here with more narrow rows, much higher density, moving towards maybe not quite the level of canopy that Stefano was talking about for fresh market apples, but we're trying to investigate how far can we push the system and still have it be profitable for cider apples. One thing I will note about the above system and from a previous presentation is you do have the ability to have more than one economic, potential economic product out of here. And here they have the sheep as well as the apples in the same piece of land. So while the trees aren't very efficient, they are double dipping in a sense. Okay, cultivar selection. Sometimes I'll use the word cultivar or cultivated variety. Other times I'll use variety. I mean the two synonymously. They have slightly different technical meanings, but I'll just use them interchangeably today. So I thought maybe, I don't know that I'll be able to see the chat, but you could put this in the chat anyway. And then when I break from the slides for a moment, I'll take a look at some of your answers. But if you are producing hard cider, what varieties are you using? What are you guys growing out there in California? I'd be really curious to know. So just pop that in the chat and let me know. Those who are engaged in the cider industry are very familiar with this classification system. This came out of Long Ashton in England over a century ago, and we still use it today. There are other classification systems out there from France, from Spain, but this is the one that I think really has stuck with a lot of American producers. And dividing apples into these 4 categories based on the tannin or phenolic content, whether it's above or below 2 grams per liter. And this is using a Lowenthal permanganate potassium titration method, and then whether or not the acidity level is above or below 4.5 grams per liter of malic acid. So once you have those 2 variables and the thresholds, then kind of divide up the apples so they're bittersweet, bitter sharp, sharp, or sweet. For the most part, for us in the US and in most regions, what we find is that there are plenty of sweet apples and there are plenty of sharp apples available, but let me see if I can get that pointer to work. Well, for some reason it's not quite working right now, but I think my pointer is working all right. But it's the ones that with the bitterness that are harder to find, the ones that have a high tannin concentration. Of course, the fruit quality, juice quality is not the only factor that is necessary. And we really have to think about all these other parameters. And people get caught up a little bit in what is a cider apple? How do we define them? And, in fact, any apple can be a cider apple, right? They all have sugar. Sugar can be metabolized by yeast and converted into ethanol. And then you have hard cider. And so I think it's really important to not just think about what we're able to mimic from other parts of the world, but also, what other varieties are possible for us to grow. Even we have producers looking at different species and hybrids, traditionally called crab apples. So when we've grown a lot of the European varieties, we've noticed lots of problems, to be honest. And Dabinet, which is the most widely grown cider apple in all of England, we've had lots of problems in our climate with cold hardiness. Fire blight, I've heard mentioned a few times in the talks today, really can be a major issue with a lot of the cider varieties because they tend to be late blooming. So fire blight, by the way, is a disease, Erwinia amblivora, that originated in New York. And so when they selected a lot of these cider apples in England, France, Spain, etc., they didn't have fire blight at the time. We were always concerned about the invasive insects, the invasive, diseases that we have here, the new diseases that we have coming into the States. But fire blight is one that we've shared with the rest of the world. So they made a lot of these selections for late blooming varieties hundreds of years ago to escape frost. But when we grow them in our climate, probably the same for you out in California, from what I'm hearing, because they're late blooming, it's warmer when they bloom, and they're more likely to get fire blight. A lot of them are also really biennial, meaning that they have an on year and an off year, and it's just terrible to think about that if you're a cider producer, where one year you've got almost no fruit, and next year you've got this bumper crop. How are you supposed to have a business out of that? It's very problematic. So I'll talk about that in a bit too. And there's some other issues here as well. So the ideal cider apple, of course, would be in the middle of this Venn diagram. I'd say the American industry is still very much so searching for that ideal cider apple. There's some candidates out there, but I don't think that there's anything that I've seen which says, oh my God, this just hits the mark on every one of these potential circles in this diagram. If we look around the world, it's a very busy slide, but just to show you, it's in the left column, it's varieties that are grown in the UK. In the middle, it's Spanish. And then the far right, it's the French varieties. I would say the UK industry, they really narrowed it down in the 80s to Dabinet and Michelin as the two that they really decided to focus on, particularly for their macro scale. They refer to sometimes as their cider factories. And that's how they really scaled up their industry. Some of the smaller producers will still use some of the other traditional varieties, your Arlington Mill, Harry Masters, Bulmers, and others further down. Both in Spain and France, you see that there hasn't been as much of a concentration of a dependency on just a handful of varieties. And they really, I think, appreciate and value the diversity that they grow to make up their ciders. And you don't see very many ciders being sold as single variety or, having even any of the variety names listed. But these are just some of the common ones that are grown in each of the regions. So as I already mentioned, this is what they narrowed their list down to in England, Dabinet, and Michelin. I'll say that they have about another dozen, making up the other 20%. And they do have releases from the Long Ashton Research Center research station. I mentioned that before. They made that classification system. They had a breeding program that ran in the 80s, and they released a bunch of varieties. They call them the 21st cider apples, sometimes nicknamed the girls because they're named after women in the cider industry. And we've actually imported the vast majority of them, and we just started to trial them in the field. We planted our first planting of them in 2025. So data from that forthcoming. What's being grown in the States, it's pretty widespread. And we did this with Carol Miles a bunch of years ago, but you can kind of see that the American producers are really just trying everything. And it's a giant variety trial happening all over the country right now. But here are some of the more common ones that you'll probably be familiar with if you grow cider apples. I mentioned the USDA apple collection or apple germplasm collection, the Malice collection, and that's a resource that we've had a lot of access to in my program. We've published a number of papers on this, doing a lot of genetic work, phenotyping. We've identified and hope, tried to resolve several misnamed varieties that were planted in there. It's some interesting things here. So thinking about that classification system of sharp and sweet and bittersweet and bitter sharp. And so it's just using those two scales here. This is titratable acidity. This is a different measure of tannins that we use in my lab, the full and shock 2. What we see here is here's some of the apples you might be familiar with, Gala, Dabinet. They do in England, they select some apples that are particularly sweet, but you'll notice that in the yellow and gray areas here, this is where a lot of the fresh market apples will reside. A lot of these red ones tend to be particularly really high tannin, but low acid. And that's a lot of the French varieties, and that speaks to some of the styles of cider they make, doing a lot of keeving. We see that some of the English varieties tend to be more on the lower end of the bitterness and even some bitter sharps in there. And we can see the Spanish varieties over here, super sharp. They have a lot of acidity, and that also speaks to the type of cider that they're making. In America, we don't really have a specific cider style yet, right? Where people are trying all different things, and there's a lot of cider that's being made and then other flavors added to it. So it's really a wide open market. Over here, you'll see that there's a lot of the species. This is Malus seversii from Kazakhstan, and hybrids over here. And so these are interesting. I don't know that you'd make a cider out of them. Maybe you could add a little bit to a cider, but we're really interested in them to look at their genetics and potential for using them in breeding programs. We have done a number of variety trials, published some work out of there, and I'll mention in a moment some of the quote unquote, ones that we're suggesting for New York growers to try. I'm not sure how they'll do in California. Your climate's different, and you'd really have to trial them there. But I do also want to note that in addition to we're trialing all these old varieties, even some new varieties, we have a lot of producers in New York, and I kind of know that there's some of this happening out in California as well. And people are actually going out and foraging varieties, from sometimes it's from abandoned orchards or old homesteads, but other times these are wild seedlings. And in fact, we did a project with some money we had from a program at Cornell, and found out that about 80% of the apples that were being foraged, or the trees that were being foraged from, were actually unique seedlings, right? They were not just abandoned trees. It wasn't just this is an old Baldwin or a Northern Spy. So I was really struck by that, because I really expected to see a lot more known varieties, and people were picking that fruit. So for New York, what are we suggesting? The one that's really risen to the top for us is Porter's Perfection. It's a bitter, sharp, fairly annual bearing, blooms more in line with Gala, so kind of more mid-season. If you can survive the first couple years and not have a challenging winter, Dabinet's a good variety. And then some others that I think are useful for juice quality, but not always for productivity. So really emphasizing porters for us. A lot of our producers also really like Golden Russet. It's a tip-bearing variety, can be a little tricky to grow, but has super high sugar, up to 20, 21 brix. We also really emphasize using disease-resistant varieties, and a lot of these typically have a lot of acid to them. So they're really nice base juice to make cider out of. Liberty, Enterprise, Gold Rush are probably the three that I think are the most reliable and also make really nice cider. In terms of rootstocks, we're still trying to figure this out. And in fact, my lab started a project just a couple of years ago. We planted 8 different rootstocks in 14 different sites around the country. We don't have one in California, unfortunately, but we do have in Washington State and some other regions. And I hope the data will still be useful for your growers there. The 2 rootstocks that I think we're leaning into right now, and again, we have a lot more work to do to really say definitively which rootstocks to use, but I would say the 2 that we're really leaning into are G890, which is about a little shy of an M26 size, and G66. which is a newer release, but I think has a lot of really nice potential for the cider industry. We also have some trials looking at tree spacing to try to figure out what the optimal tree spacing is, and I'll mention that again in a minute. On the far right here, you'll see that there's this picture of this Porter's Perfection apple. They just have this unique anatomical feature where they'll have this fused fruit called polycarpy. Doesn't really much matter because it's going to be milled and turned into juice, but it's just kind of an interesting oddity. Okay, orchard design. If you go to England, you'll see cider apple orchards, and true in France and Spain too. Again, grazed orchards on this high trunk, very low density, extremely low density compared to what Stefano showed. Some of our early pioneers in the cider industry in the US really looked at this and thought this was the way to go. I'd say be cautious about it, right? Because this is going to take a long time for the tree to come into bearing. These orchards are really meant to be, 50, year investments. If you're a new business and you want to start producing fruit in the next couple of years, this sort of system, isn't going to get you there quickly enough. Even in England, though, they've moved to higher density systems, 8 by 18, spacing some 6 by 16, 300 trees per acre. They're still using very big semi-dwarf trees, but a little bit more what you'd expect to see from, a traditional orchard in rows. In New York, a lot of our producers are really leaning into what we've learned from the fresh market industry. So we have a lot of producers trying tall spindle systems. This one is at 3 feet between trees, 12 between rows. Michelin on the left, Dabinet on the right. This is in along Lake Ontario in New York. And you can see a tremendous crop load in there, just really a lot of fruit, these are in their fourth leaf. So just some more pictures of some different orchards in New York and what it looks like. And you'll notice fruit on the ground, and I'll get back to that in a minute. If you are from an area where you can support higher density or have the knowledge and know how to do multi-liter trees, we are seeing some of that happening. This is a cider orchard in Washington State, taking the lessons that Stefano and they've learned from their fresh market and trying to apply it to the cider apple orchards. Okay, so what's actually profitable, right? Where is the profit line here? So this is a net present value calculation. At 0 is your break-even line. So, we're looking at case studies of 6 different orchards in New York. And of course, right at the year of planting, there's more cost than there is return, that you're making money. You're not selling fruit for several years. And what we found is that for 5 out of the 6 orchards that we modeled, they were profitable. They were making money selling cider apples. But a really wide range here between the most profitable and the least profitable orchards. And some of that was due to, pause the sound there for a sec, some of that was due to the establishment costs. When the growers were really leaning into the fresh market systems, they were not able to recoup the cost at the price that they were receiving for cider apples. And then also, some of the orchards are in areas where our labor costs are super high. And that's what we're showing here on the far right there is hand harvested versus machine harvested. And I'll just say that when we looked at that, and I'll mention that again, that mechanical harvesting is really critical component to how I'm thinking about cider apples of the future. Okay, another question for you guys. If you have cider apples, what are you doing for crop load management? What are you doing for thinning? Are you doing it by hand, doing chemical? Just pop it into the chat. Give me some feedback there. We've done a number of studies on this. I mentioned earlier that a lot of the cider apples are biennial. They have an on year and an off year. And so we were trying to manage that. But first, before we try to manage it, the first thing that we tried to do is figure out what was the ideal crop density, how many fruit per tree is necessary for a cider apple tree, right? So we know it's probably not going to be the same as a fresh market apple. Fresh market apples, you're really concerned about that fruit size and that color. But for cider apples, we can probably crop a little heavier. So this is just first looking at return bloom, how many flowers are on the tree the season after. We have the crop density on the x-axis here. And in the red here, we can see the point of no return. And so this is what we see for a lot of cider apples, that when you get to over 15, and then certainly for some of these that were over 30 fruit per trunk area, centimeter squared. It's kind of a funny metric, but it's the it's the area of a trunk. So we measure the circumference, and then we calculate the area of the trunk. It's just an easy way for us to, as scientists, to try to equilibrate across all the different sizes of trees to come up with how many fruit per tree should we have. At 10 fruit per tree, we're talking about, for a spindle type of tree, about 200 apples. And so we use that information, and then we also looked at the same metric at the bottom, the crop density, but some of the important quality attributes for cider. So phenolics, the soluble solids, the bricks, the acidity, and the amino and nitrogen content. And we can see as crop load increases, we have these declines in fruit quality for all of these metrics. So we use that data from the previous slide and from this data here, put them together, and came up with this okay, let's start at this point, 9 fruit per trunk cross-sectional area, about 200 apples per tree is really what we should be looking at, where you're going to balance getting enough return bloom and also have the fruit quality where it's not so low, right? And if you let your trees become biennial, you're going, those on years, you have a lot of fruit, but you have a lot of fruit of lower quality for cider making. Some of our growers tell us that, oh, you can't chemically thin them. They're too difficult to thin these cider varieties. We did some research on that and have shown that, no, that's not true. And I won't go into all the details here, but I have a master's student who's going to be finishing up the project this year. So hopefully we'll get it published sometime this fall, and we can share more of the data about our treatments. But you can chemically thin cider apples. And we found that you get a 60% reduction depending on the treatment you used, which resulted in almost a 300% increase in return bloom. Okay, so for nitrogen management in cider apple orchards, this is really important. Those who are in this or familiar with the wine industry, you're familiar with this concept of YAN, yeast assimilable nitrogen. For cider apples, or apples in overall, there's very little ammonium, which means that most of the YAN is actually just primary amino acid. It's just the organic forms of nitrogen that are important. If your nitrogen level is too low, what happens is that you get reduced sulfur. Can smell like, skunky smells or natural gas smells. Actually, they add that to natural gas so we smell it. But that's what the yeast do. So we've done some different work here to show that whether you apply nitrogen to the ground or you do foliar nitrogen applications, in this case with urea, you can significantly increase the juice nitrogen level. Okay, so this can be done in the field. You don't have to do it in the cidery. You can do it in the cidery, but you don't have to. We found that one of the interesting things, though, is that most of that increase in YAN is coming from amino acids from the organic form. So that might be of interest to those who want to get into yeast metabolism and really want to talk about some of those more nuanced things. But we also found that it was by doing the nitrogen applications in the field, we were able to significantly reduce the hydrogen sulfide, the stinky egg, skunky, natural gas smells. We found that it was more cost effective to apply in the field than to using some of these expensive proprietary products like Fermate O. Of course, diammonium phosphate, or DAP, is the cheapest product and cheapest way of handling it, but it's it's like synthetic nitrogen. It's not always what all the cider producers are wanting to do for their nitrogen additions. And then additionally, when we did these additions in the field, we found that the nitrogen content, by increasing the nitrogen content in the by applying nitrogen in the field, we were able to significantly increase a lot of the positive aromatic compounds for cider production. So that was really exciting to us, that not only reducing the faulty smells, but we're also increasing the positive attributes. Okay, and then just real quickly on harvest management. Again, a question for you if you want to pop anything in the chat. If anybody's using mechanical harvesting, let me know. What we found in our studies is that regardless of your scale, as long as you choose the right machine for the scale of your operation, it's cost effective. And so we modeled this a number of ways. We have the publication there. I can share it with you and share it with Ellie, and she can share it with you. But it makes sense, right? It's a harvest is a very expensive operation. And there are some small scale versions out there. This is one out of Austria, and this only costs $6,000. You could have several of them maybe running through your orchards. It just kind of picks up the apples. It runs off of a battery that is used for e-bikes. So a cool little tool like that. For the sake of time, I'm not going to go into all the details, but you can also manage your harvest timing with using different plant growth regulators. And I think this is something that, as our orchardists here in New York and other places get larger in scale and really trying to dial in their management, these are just some of the different tools that we're providing. Of course, for ground harvesting fruit, you have to really think about the process stream. You're not going to be able to store that fruit, and you're going to have to really get it pressed within days of harvest. We also have to worry about enteropathogenic organisms, and but really, there's minimal risk. There's never zero risk, but there's minimal risk from ground harvested fruit. And in fact, when I mentioned that, it was part of a study, and we did find that a new species, newly discovered species, could produce patulin in cider, regardless of how we fermented it, and even when we did things like autoclave it. It was a really hardy thing. So something that we're looking out for. It's not widely present in our soils, but it's something that is out there and natural that we're finding. But I will mention that we worked with Angry Orchard quite a bit on different projects, and they are actually producing a cider that's 100% using drop fruit. So they're really testing the market for us in the US. Okay, so just to wrap up here, what's the future? I think we're trying to narrow down the cultivars to use for cider. It'll be interesting to look at the chat in a minute. I think looking at medium density orchards, 4 feet by 3 to 15 feet, trying to use some trellis, but really minimal trellis. I'll mention again the 2 rootstocks that I think are worth investigating, and then also looking at mechanical hedging and harvesting. So just some pictures of my research program and team and different funding sources. Okay, so I'll just break there, and maybe I'll pop into the chat real quick, and then we'll let everybody end their program in a moment. Oh, I think I have to stop my share. See that? Yep, there we go. Great. Hey, Ellie.
So much. That was really fascinating. Yes, we do have a lot of comments in the chat.
Yeah, great. So, yeah, just some of the varieties that we see similarly out there. I see Hughes mentioned. We just published a paper. A lot of the hues that people are growing that came out of the USDA germplasm collection are actually not the historic hues. It's a Virginia crab. It's a different variety. Just a note. Yeah, so it's really cool to hear about some of how things are happening out there.
Yeah, and David Klein mentioned this in the chat, and I mentioned it briefly in my presentation too, but and actually, you probably know this because I didn't realize it, but you've spent time out here in the past. But a lot of our apple orchards do have tons of different rootstocks, different scions, packed into one orchard. So it's very common to have, yeah, 80 to 100 cultivars in one orchard, which is pretty mind blowing to me. But so yeah, thanks, David, for mentioning some of those
It
specific
can be it's a very difficult thing to manage, but it's not uncommon in other parts of the world either.
Oh,
But it's there's a trade-off. You get a lot of diversity, and hopefully something survives every year for your cider production. It's hard to make the same cider year in, year out when you have that diversity, and it's also really hard to decide when to do different management things in the field.
yeah. Laura has a question in the Q&A if you'd like to check that out.
Um, Curious about the idea that nitrogen increases the positive aromatics. Does that come out in the fresh juice or only during fermentation? So it's we found it in both. The yeast, of course, are going to have a huge influence about which esters are there during in the actual finished product. But we did find those increases in the aromatics in both the juice and in fermented cider. And we did it through a sensory panel as well. And our sensory panels were able to differentiate the different nitrogen treatments.
Very cool. Thanks. Any other questions? I'm looking through the chat to see if I missed anything. Let's see. Give folks one more minute. You probably know this already, but we do have the Gravenstein Apple Fair here, which is our big annual, agritourism moment for apples. I feel like, although there's lots of programming throughout the season, getting folks out onto apple orchards and familiar with cider and stuff like that. But was that going when you were here?
Yeah, that was going, and yeah, I mean Gravenstein apples. I love Gravenstein apples. I mean, they make nice cider early, but I think they have a nice acidity to it, nice aromatics. I know a lot of them are getting a lot of those old Gravenstein orchards are getting ripped out. That was happening many years ago too, being pulled out for Pinot and Chardonnay. But those who hold on to them, they are a really nice historic apple.
Yeah, that's true. Acreage unfortunately has been declining for a while now. But we also have growers, like folks in the chat who are planting new orchards, apple orchards, which is exciting. I think seeing new apple orchards here is very exciting to me. And we have some folks who are maintaining those older plantings and thinking, creatively about what to interplant when they do have trees that go down or sections that they want to replace too, which is always nice to see. But yeah, so thank you to all of the growers here who are keeping the apple industry alive. I appreciate it. We all appreciate it. And yeah, it looks like we don't have any more questions, so I think maybe we'll wrap up. I did really appreciate your offer, Dr. Peck, earlier at the beginning as well to come out here. I know several farmers who are here right now who are probably cheering, saying, "Yes, Ellie, like ask him to come out, take him up on that." So yes, please know
Great,
that
let's make it happen.
that'd be
I've got
great.
family in San Francisco, so
Oh,
it's an
cool.
easy ask.
Oh my gosh, wonderful. Okay, great. Well, we'll be here. And yeah, we can follow up about that another time. But yeah, thank you so much for everything, for sharing your knowledge and supporting our Apple folks here.
Great. Have a great afternoon, everybody. Hope to see you soon.
Thank you. Have a good evening. Bye, everybody.